Good morning, everyone. Hello, good morning. We're going to go ahead and get started. So if you want to start taking your seat, welcome everyone to the Rare and Orphan Disease Conference. We're very excited to have you here with us today. I just want to go over a few housekeeping items. Just a moment. Maybe. Okay, here we are. So just a couple of housekeeping items before we get started. Everyone has received their name badges. On the back of your name badge is a QR code that has the agenda for the next couple days, as well as the Wi-Fi code. We also have those placed throughout the conference center at various stations, so if you need it. Also, all CPATH staff is wearing a little CPATH pin, so if you see anyone with this pin, if you have any questions, flag them down. If we don't know the answers, we'll find it for you. So on the agenda, you can see we have a couple breaks throughout the day. So we have a morning break, we have a working lunch, an afternoon break, and then we have a networking event this evening that we hope that you'll join us for. Also, just wanted to let everyone know that today's main sessions here in the ballroom are being recorded. We will make those available on our website after the event. We will also have, you may see a photographer and videographer walking around today here from CPATH just collecting some general footage. So just wanted to let you guys know that. Also, for those who haven't seen yet, if you go out to the main lobby, past the elevators to the left, those are where the restrooms are. So if you are looking for that or flag anyone down again that has a pin, they can help direct you. And then just a quick reminder to silence your cell phones in here. And then also wanted to let you know that we have, there's several power strips, outlets out in the main area. There's tables that have plug-ins. So if anyone's looking for somewhere to charge a device during breaks or anything like that, there's lots of places around to allow you to do that. So again, if you're having a hard time finding anything, just let one of us know. And with that, I would like to go ahead and welcome up to the stage. There we go. So a very warm welcome to Dan Jorgensen, who is the Critical Path Institute CEO. So welcome, Dan.
Thank you. Thank you. It's a pleasure to be here today. My name's Dan Jorgensen. I'm the CEO of CPATH and a pediatrician. And on behalf of CPATH, I want to extend a warm welcome to Washington, D.C., and to this meeting. But first, on this important day, we should take really a moment of silence in memory of those affected by the events of September 11th. Can you hear? So again, thank you. Thank you, everyone, for attending this meeting. And we do have quite a really good representation here of researchers, clinicians, scientists, providers, regulators, and representatives of the biopharmaceutical industry. But in particular, I want to thank the patients and their families for being here today. You are the ones who endure the hardship of a rare disease on a daily basis and are hoping for new therapies. We admire your courage and resolve. Thank you for being here. Over the next few days, we will discuss the many ways that we can improve and accelerate the drug development process so that important medicines are available to you, to all of you, especially the patients, as quickly as possible. As you know, drug development takes way too long, up to 10 to 15 years in some cases. At CPATH, we accelerate the drug development process, we remove the bottlenecks, we make it more efficient for everyone. We are unique because we provide a neutral space where pharmaceutical companies, regulators, and all the people that I just mentioned can come together, and then we integrate the information we've received and we generate actionable solutions. This way, drugs can be approved faster, which improves the health of people worldwide. And this is reflected in our mission statement, which you can see on the slide. Basically, we advance drug development, we improve lives together. That's our new tagline, and that's what you'll see when you go to our website. But especially at the bottom there, you see the mission statement. We lead collaborations that accelerate drug development, advancing better treatments for people worldwide. As a neutral convener, we lead collaborations, which you can see on the left-hand side of the slide, and we try to do so, and to be effective in doing that, we continue to bolster our core competencies, which you see on the right-hand side of the slide. Together with our partners and collaborators, many of whom are in this room, we can address unmet medical need across many disease areas, including rare disease, as seen on the right-hand side of this slide. You can see all the different disease areas that we're involved in now, and we hope to be involved in even more. In response, CPATH and partners, including our colleagues at FDA, have developed a critical path for rare neurodegenerative diseases, also known as CP-RND, linking CPATH's core competencies and the Rare Disease Cures Accelerator Data Analytics Platform, also known as RDCA-DAP, with the relevant consortia and concentration areas, as seen on the right-hand side of this next slide. Importantly, if you see on this slide on the left-hand side, really the burden and unmet medical need of rare diseases. We applaud everyone's effort involved in CP-RND, and as part of this meeting, you will hear about the impact and highlights of the first year of CP-RND. But it's not just a medical or public health burden, it's also an economic burden, as you'll see here. Basically, it takes a lot of money to develop these drugs, but it also is a huge burden on families, people who need the therapies. We hope and believe that through our efforts on the health side of rare disease, we can also make a difference on the economic side. In other words, it's not enough to develop and approve new therapies. We need to ensure that patients with rare diseases have access to needed therapies once they are approved. A lot of progress has been made, but a lot of work remains. We hope that this conference is a catalyst for important work in the area of rare disease. And here's the list of conference objectives. Again, welcome to Washington, D.C., and welcome to this important conference. Thank you for being here, and I will now turn the podium over to Dr. Celia Witten of FDA. Thank you so much.
They're getting mic'd up. Maybe I can give you a little bit of orientation for today. I'm Colin Hovengay. I'm the Vice President for Rare and Orphan Diseases at Critical Path Institute. Day one is obviously action-packed, as you can see from the agenda. We're going to walk through opening our meeting with FDA, giving us a fireside chat. We're going to have a second session on from industry, giving their experience in rare disease drug development. Then we're going to talk about the initiatives and collaborations between FDA, NIH, FINH, and CPATH to launch the ALS initiative and the Act for ALS activities. Then we had a little bit of a snafu for the DHT discussion. For sake of time, we're going to omit that for the 10:45 session. Then we're going to move into rare disease endpoints with FDA, and then for the showcases this afternoon, the midday, we've given you a little bit of time. You're going to get lunch, and then you're going to head to the showcases. There are sessions that are down here, and there are some sessions that are upstairs, and so make sure that you connect with someone with a pin to make sure you orient to the right place. Then in this afternoon, we'll come back here and convene again, and we'll have a series of sessions on clinical trial simulations, unveiling our COA web resource. We'll have one with Sergey, who's our ED for sickle cell, talking about endpoints again. And then Trina will wrap up the sessions in here on disease staging with Huntington's disease. We'll have a second round of showcases. So again, if you can't find your room, grab a CPATH person, and we'll come here for close with opening remarks. And then later this evening, we'll have a breakout session, or a breakout and networking opportunity where the reception where you can meet and greet with many of your colleagues. So we have everybody up here. Okay. Thank you again for everyone for joining this session. You'll see many people show up. The travel feel free to come and go as you please, but the travel has slowed some people from getting here. So for the first session, I want to have them introduce themselves. We're going to have an FDA Fireside chat looking at the impact of rare diseases and FDA's perspective on them. And they're going to convey some of the programs that the FDA is undertaking, and this will be followed by a Q&A. Really exciting activities going on in agency, and we want to make sure to introduce those with the team. Who might be introducing slides first?
I guess I should start, yeah. Do we have a clicker? Okay. Perfect, thank you. All right, good morning. I'm Jacqueline Corrigan-Karai. I am the Principal Deputy Center Director in CEDAR. It's really a pleasure to be here. I'm not sure if I'm getting an echo because I have 2 mics on. Am I okay? All right, good. So what I'm going to do is just talk a little bit about the landscape for rare diseases in CEDAR, and then I'm going to hand it over to my colleagues to talk about some more specifics. So here we all are, and I'll have each of them introduce themselves as they come up. So these are just some numbers for you, and it really shows you the CEDAR orphan drug approvals of new molecular entities by calendar year. The last year in 23 is only through September 6th, so a little bit lower. But you can see from 44% to 52%, but on about half of all our NME approvals are for rare diseases, which is really encouraging. I know there's more work to be done, but certainly. And importantly, many of them are able to take advantage of what we call our expedited programs for development, including priority approval or priority review, actually, which really means that when it comes into the FDA and we have preliminary, we can say that there's going to be a significant improvement, that we can rapidly accelerate a little bit the review of that package. Fast track is something that you can get based on preclinical data as well as clinical, and then breakthrough therapy is when you have preliminary clinical data that shows that there may be a substantial benefit over what's available. Now, as you can see, about half of them get either one of those. Accelerated approval, of course, is a little bit lower, and that's really because you need a biomarker. You need a biomarker that we say is reasonably likely, so that's one of the limiting factors in that one. So one of the things to remember, we have a number of initiatives that are regulatory science initiatives, and they span, they're not always rare disease specific, but they can be incredibly important for rare disease development. For example, complex innovative trial designs, when you're thinking about designs for smaller trials. The critical path innovation meetings, if you're thinking of something new, you want to get some early, this is not a pre-IND, this is really to discuss your ideas with us. Model-informed drug development, and then, of course, patient-focused drug development. I think many of you may be aware of. Many of those sessions have been around rare diseases, and now I believe all 4 guidances are out so that you can really think about how to integrate the patient perspective in drug development. We support RDCA-DAP, very important, getting that data together, getting it in a structured manner that we can really learn from. And I'm going to talk a little bit about the last 2, which is real-world evidence and digital health technologies. We often think of real-world evidence as big data, but I think it's really what real-world evidence is trying to use every piece of data that we have from clinical care and see if we can apply those learnings into the regulatory. And then, of course, digital health technologies. So in the real-world evidence program, we've put out a number of guidances, and I think I'll highlight several that are probably quite relevant. One is our externally controlled trials for drug and biological products put out just this year. Now, I'm not saying that every rare disease development program should be an externally controlled trial. We often need randomization, but just if you are going to be taking that approach, this really gives you some important guidance on what to consider in terms of what kind of data, the comparability issues you have to think about, analysis considerations, and then considerations to support regulatory review. The other guidance I just want to highlight, which was done a couple of years ago, and we're finalizing it right now, is the registry. So, again, registry is quite important. And so how should you think about them and using them for regulatory decision-making? How should you design them, actually, is in this guidance when you're thinking about that. And then how do you do linkage, and what do you have to think about when you're trying to link a registry to outside data, EHRs or digital health technologies, and then how we might review it and kind of thinking about what data needs to be submitted to us. So I think those are important guidances to be aware of. The other thing is there's been a lot of attention on digital health technologies. We published a guidance on this, I think it was almost 2 years ago, that we had a number of comments, hundreds of comments. So there's just great interest in the use of digital health technologies. And then under PDUFA 7, we have a commitment, and we published the framework for the use of digital health technologies in drug and biological development just this year. And so this is really thinking about how do we change trials? How do we think about collecting endpoints, make trials more accessible or different endpoints, really understand the patient's journey outside of perhaps their visits. And that really gets into thinking about, can we do trials where at least some or even all of the trial-related activities take place? Again, that'll depend upon where you are in your development program, what's the intervention. But thinking about how do we make trials more accessible. And towards that end, we just published several months ago the guidance on decentralized clinical trials for drugs. And so really thinking, is it video and telemedicine visits? Is it direct distribution of the product? How do we make electronic informed consent more easy, home visits, and of course, integrating digital health technologies. So all of these are, innovations that, can be used in the rare disease space, especially when you're thinking about diseases where, geographic recruitment may be quite wide. So this is just an overview of some of the regulatory science initiatives that are not rare disease specific, but certainly can be leveraged by the rare disease community. And now I'm going to turn it over to Carrie Jo, who's going to speak a little bit more about our newest program that is quite specific for rare disease.
Thank you so much. Hopefully everyone can hear me without an echo. Sounds like it. Thank you. Sorry, I'm just trying to advance this slide.
The green one, the big green one.
Did pick green. Oh, big green. Now we'll go back one. There we go. So back in May of 22, CDER really wanted to consider how can we harness all of our collective experience and collective knowledge to really try and accelerate rare disease drug development from the regulatory perspective. And to that end, we decided to launch the Accelerating Rare Disease Cures Program, or the ARC program. The vision of this program is that we could speed and increase the development of effective and safe treatment options to address the unmet needs of patients with rare diseases. And the mission for this program was really to drive the scientific and regulatory initiatives and innovation and engagement to accelerate the availability of treatments for patients with rare diseases. As you saw from the previous presentation, this represents a substantial degree of the work that we do at the FDA when it comes to new molecular entities and programs. So what does the ARC program look like? Well, it's a program that has come together and is governed by senior leadership from the areas of CDER that are most directly involved with regulatory drug review. So that would be the Office of New Drug that houses your clinicians and your pharm tox representatives. That has the Office of Center Director, where you have senior leadership, such as those who are speaking with me today, as well as the Patient-Focused Drug Development Program. And then you have the Office of Translational Science, which houses our statisticians, our clinical pharmacologists. And bringing all of these senior leaders together to govern the ARC program and look as an umbrella program over those initiatives that are specific to rare diseases, as well as those that can inform rare disease drug development, was critically important. The program is managed by the Rare Diseases team, and I realize I did forget to introduce myself. I'm Dr. Carrie Jolie. I'm the Associate Director for Rare Diseases in the Office of New Drugs, and I lead the Rare Diseases team. Our team is PDUFA mandated to carry forth and connect the rare disease programs for CDER. Now, when you look at CDER's ARC program, you have the ARC governance board and you have the rare diseases team which manages it. But it doesn't stop there. It is an umbrella program for the center. And therefore, we work very closely with our colleagues across other offices, such as the Office of Medical Policy, which has a lot to do with benefit risk and real-world data and real-world data and real-world evidence and digital health technologies. You have the Office of Surveillance and Epidemiology, which additionally is informative to our use of real-world data and real-world evidence, as well as post-marketing safety. You have the Office of Strategic Programs, which is where a lot of the benefit risk work is centered. And then you have other offices, CDER offices and divisions that may contribute to all aspects of rare disease drug development for subject matter expertise as necessary. And then we also work closely with other FDA offices and programs. So that includes our good partners at the Center for Biologics, Evaluation and Research. We have many similar indications and conditions to manage, and we work with them very closely. We have a Center for Devices and Radiological Health. We work with the Oncology Center of Excellence. We work with the Office of Commissioner, specifically the Office of Orphan Product Development very closely, as well as the Office of Pediatric Therapeutics and the Patient Affairs staff. So ARC is really an umbrella that pulls together all of these groups in order to really drive our focus to develop methods and innovation to aid rare disease drug development. And I have our website on there, which I encourage you to go to. So what are some of the things we really focused on in our first year of development? This was really a focus on engagement. It is critically important that we share our regulatory thinking about the challenges and conditions that affect rare disease drug development. And to that end, there were 4 workshops held under the ARC umbrella the first year. The first one was in conjunction with our partners at the NIH in regulatory fitness for rare disease clinical trials. And that was a conference that spanned many, many aspects of drug development and lives on in perpetuity at our website, as well as the FDA Duke Margolis Virtual Public Workshop, which we had on translational science development, specifically on surrogate endpoints, biomarkers, and more, which are critical to rare disease drug development. Many of these conditions are slowly progressive, and to try and measure a clinical endpoint during the duration of a trial can be extremely challenging. So this conference really focused on how to develop those endpoints, what are the challenges, and what are some potential solutions, as well as examples. We additionally held a CIRSE in 2023 of May, and this was one that was very, very timely. It had to do with addressing challenges in design and analysis of rare disease clinical trials, specifically focused on the use of real-world data to inform the design of rare disease trials, as well as challenges in analysis with these small populations, so statistical challenges. And finally, there was another CIRSE held with the University of Maryland on quantitative systems pharmacology and some of the more advanced modeling that can be done. Additionally, it's incredibly important that we engage with our patients. So we engaged in 23 rare disease patient listening sessions that first year, held between patient affairs or the professional affairs and stakeholder engagement, and 19 patient-focused drug development meetings. So this is just a way in which we are trying to engage more and listen to our patient stakeholders about what is important to them about their conditions that can help to inform rare disease drug development. And then finally, I'm just going to end with a plug for our quarterly newsletter. We are trying to get the word out as much as possible on rare disease happenings that the FDA is participating in. So I would ask that everyone go through and sign up for that, and you'll find out about things. All of the workshops that I mentioned are on our website. They live on in perpetuity so that people can go and educate themselves on different aspects of rare disease drug development. And now I'll turn it over to Dr. Baracchio.
Green, okay. Hi, I'm Dr. Teresa Baracchio, and I'm the Acting Director of the Office of Neuroscience in CDER. And I'd like to take some time to discuss 2 rare disease actions that we've had this year that have been impacted by some of the initiatives described by Dr. Corrigan-Kurai and Dr. Lee right before me. The first was the approval of omaveloxolone, which is also known as Skyclarys, for the treatment of Friedreich's ataxia. And this is the first treatment that's approved for this disease. Friedreich's ataxia is a neurodegenerative disease that begins in childhood and has a very heterogeneous course and causes impairment in motor function. So the approval was based on substantial evidence of effectiveness based on a single adequate and well-controlled study with confirmatory evidence. The sponsor conducted a adequate and well-controlled 48-week randomized controlled trial that had a clinician-reported outcome that was positive, although there was not strong support from secondary endpoints. Although the study was positive, it was not strong enough to stand on what our usual standard would be for a single trial approval alone. So we do have a pathway that's available to us where we can utilize confirmatory evidence that would substantiate the findings seen on the adequate and well-controlled trial. So we worked with the sponsor to identify confirmatory evidence that could support the primary evidence from the controlled trial. When we work on these programs, we do take into consideration the rarity of the disease, the unmet need, and the challenges in the ability to conduct a second trial. And we felt that it would be quite challenging for the sponsor to conduct a second adequate and well-controlled trial in this disease. So we were willing to work with them to try to figure out a path forward. And looking at the confirmatory evidence that the sponsor proposed, they had proposed to use mechanistic data to support the drug's mechanism of action that was relevant to the disease pathophysiology. They conducted a delayed start analysis using the open label extension data compared to the controlled period of the study. And they used, took their open label extension data and they compared it to a natural history study, which was the FA-COMS. No single element of these was served as a piece of confirmatory evidence alone. We looked at this as a total package, and I think that's one thing when you look at our guidances. We have discussion of confirmatory evidence in the 2019 effectiveness guidance. And when you look at that, you might be led to believe that you need to have a single piece of confirmatory evidence, but really we can look at multiple components for confirmatory evidence, and overall we put a package together that we think is compelling to help substantiate the effects seen in the primary study. I will say that much focus has been put on the open label extension compared to natural history comparison, and I've seen a lot of comments about that in the press, but I will just say, although we did utilize that data, and I don't want to downplay that, but that was not the primary evidence of effectiveness. The primary evidence of effectiveness was still a positive, adequate, and well-controlled trial, but we were able to leverage the open label extension data, the natural history data, to help support that approval. And I'm not going to go into details of that now because I believe I will get an opportunity to talk about this more tomorrow at another meeting or another session. So I'll save the details on that for that meeting. The second approval that I want to discuss is tofersen, which was an ASO that targets SOD1 protein. And it's also known, the brand name is Qalsody, I guess. And it's approved for ALS due to SOD1 genetic mutation. So this program received accelerated approval based on reduction of plasma NFL neurofilament light protein, which was found to be reasonably likely to predict clinical benefit. A phase 3 study was conducted that was an adequate and well-controlled study, and it was a fairly large study for this population. It enrolled 108 patients, and overall it's estimated that there's just about 300 patients with this genetic mutation in the US. So 108 patients is a large study for this population. And they treated patients for 24 weeks. The study at the 24-week or 28-week was the analysis point. At the 28-week point, the study was negative on clinical endpoints in the controlled study, but robust reductions in NFL protein and SOD1 protein were seen at that point. And when you looked at the open label extension data, there were notable trends for clinical benefit if you looked out longer to a 48-week period. So it was enough to suggest that this 28-week trial was not a negative study per se, but rather a failed study. And there were reasons to think, to see why the study failed. One being that it was a small study and likely underpowered for the clinical endpoint, and also that the study duration was too short. So there were reasonable rationales for why this study would have been considered a failed study and why there may still be evidence of effectiveness for the drug. And also, it was a targeted therapy that was known to impact the underlying pathophysiology of the disease. So based on this, we felt that the data was not sufficient to support a traditional approval, but that with additional analyses of NFL, that the reduction in NFL in the context of a targeted mechanism and SOD1 reduction, that NFL could be considered reasonably likely to predict clinical benefit. And so I just want to emphasize again that it's important to note the context of this the use of this NFL, which was that this is a population with an identified genetic target, that the drug targeted that genetic mutation, it showed target engagement with SOD1 reduction, and that the NFL reduction was felt to be a likely downstream effect of reduced neurodegeneration related to the disease. So that context is very important because, again, one of the things that shows up in the press is we've used NFL as a surrogate endpoint, and are the doors wide open for NFL? I think NFL is promising, but it does really need to be considered in the context of the disease and the development program and the therapy. So we're not ready to open the doors completely for NFL, but I would just say it's quite promising, and we think it should be considered as an exploratory endpoint in many of our neurodegenerative disease programs. And one other caveat before I step away was that the other thing that I've seen a lot of press about with this approval is that we approved a drug with a negative study. And so I do want to emphasize that it's not typical that we approve drugs with negative studies, but if there is a strong rationale for why a study failed and you have a strong scientifically based hypothesis to support the analyses that are done in the case of NFL and SOD1 reduction, that we can still look at the data. This is not a fishing expedition or cherry picking of data. If you have a really strong scientific rationale behind it, that should be pre-specified before you start doing your post hoc analyses. But I did want to just comment on that, as I know we've seen a lot of comments on that in the press. So I think with that, I'll stop and turn this over to Dr. Celia Whitten from CBER.
Thank you. Can you hear me? Yes, okay, good. Yes, thank you, and thanks to the organizers for inviting me to speak. I'm going to talk about rare diseases at CBER and a couple of initiatives in CBER for rare disease development. But first, because I think this audience may be more or less, some of you may be less familiar with what products are regulated by CBER, the Center for Biologics at FDA. I thought I'd just start with this slide. Allergenics, blood products, tissues and cellular products, gene therapies, vaccines, xenotransplantation products, certain devices, and then some other miscellaneous categories. And I think to this audience in particular, gene therapies is important, although some of these others are also for rare diseases, especially, for example, xenotransplantation products. So just keep that in mind too, but most of this topic will focus on gene therapies. So we do have a rare disease program at CBER, formally with the lead for the rare disease program. Some of you may know Julie Valancourt. I don't know if she's here today, but obviously it's smaller than CDER's because we're relatively smaller. And we do a lot of active collaboration with outside stakeholders and collaboration with our colleagues at CDER on projects and on policy development. But we have our own policies specifically for some of our products that we're developing too, and I'm going to talk about them a little bit later in this talk. We also have a patient engagement program with the lead, Karen Jackler, who's actually attending this meeting with me today. but again, it's a smaller program than what is at Cedar. So the Office that regulates cell and gene therapy and some other products too is actually been reorganized and is now called the Office of Therapeutic Products. But this slide is from when it was called the Office of Tissues and Advanced Therapies. And I'm just showing this slide to give you an idea of the explosion in interest in cell and gene therapies, which mostly drives this over the last couple of years. And you can see for yourselves, I don't really need to read the numbers. The blue bar represents the investigational studies, and the orange part of the bar represents continued access programs or access programs. So I mentioned that we have some specific policy development that we do or need to be working on in the cell and gene therapy area. And PDUFA 7 gave these specific obligations. So in the area of patient-focused drug development, we have completed this one. We had a public meeting November 15th on patient perspectives on gene therapy products and issued a summary report on that. Novel approaches to development of cell and gene therapy. This requires draft guidance on evaluation of efficacy in small populations, and also separately a question and answer draft guidance on FAQs from sponsors. I think these two are really going to be of a lot of interest to the public. The first one is just exactly what it sounds like, and as you would expect, there are certain differences in development for different kinds of technologies, and certainly for gene therapy, it's appropriate to have a draft guidance that focuses specifically on cell and gene therapy in small patient populations. So we're actively working on, at least at this time, thinking about the scope of that guidance. Question and answer draft guidance on FAQs from sponsors. So many of our products are quite novel, but after a product is in scientific development for a while, and there are other products like it, we may have a lot of questions from sponsors which we have answered enough times that we can give, it would be useful to the public for us to give that answer to the public. And so have a project where we're looking at all the kinds of questions and answers we've been asked over some period of time to try to develop this guidance. So that is an active development. And then this next requirement has been completed. We held a public meeting on methods and approaches for capturing post-approval safety and efficacy data on cell and gene therapy products. The other obligations are to update the guidance on expedited programs for the development of regenerative medicine therapies, including, for example, the RMAT designation, and also, I think, really importantly, leveraging knowledge, a meeting and draft guidance on leveraging prior knowledge and public knowledge across therapeutic contexts. This is something that certainly comes up. I know it comes up across FDA, but it certainly comes up specifically in the cell and gene therapy area, meaning, what have we learned that will help us develop future products? Obviously, for an area like cell and gene therapy, that's critical, both for the clinical development part of it and also for the CMC and preclinical development. So now I'm going to move on and talk about one regulatory pilot that we are undertaking at CBER that will be announced later in the year, or at least implemented, I hope, later in the year, originally called the Communications Pilot, or Operation Warp Speed for Rare Diseases, but I think it may have some other name by the time it gets announced. So Warp Speed was the term used for development of the vaccines, which were really developed for COVID in a very quickly, given how complicated an enterprise it is. And so the question is, can we similarly speed up the development of product for rare diseases by taking a look at how we do things? And one of them is communicating with sponsors. So this gives you the idea of the history and the purpose. The products eligible will be products for life-threatening rare genetic diseases showing promising efficacy early in development, And the benefits of the sponsors will be an initial meeting followed by ongoing informal interactions via email or live meetings on an as-needed basis. So you'll hear more about this later in the fall. The last two things I want to mention was we have a very active stakeholder engagement program and a very active program for regenerative medicine education for more aimed at patients and families. Although both of these programs are extremely widely attended, and we have, for the stakeholder engagement, the virtual town halls, which are really aimed at developers, we have a lot of patients attend, and vice versa for the regenerative medicine education series. So these are launched in September 2022, and they engage with product development stakeholders in a question and answer format to provide regulatory information to advance drug development. As I said, we've had really overwhelming attendance. And for some of them, and we get questions during the event, some of them are pre-submitted. To one of them that we had in April of this year, there was such an overwhelming interest and such great questions that were asked that we had a meeting in April on gene therapy, CMC, and controls. We basically had a second one on CMC cell therapy, manufacturing controls, and then more recently one on preclinical development. So I think that's, I'm not going through the slides that list what all the different events are that we had, but if CPATH posts these, I have links included at the end of the slides. They're easy to find even if the slides aren't posted. So then this is the second educational series that I mentioned, Regen Med Ed, Patient Engagement in Regenerative Medicine. And this is an educational workshop and webinar series to engage patients, caregivers, and advocates to discuss regenerative medicine therapies, explore opportunities for stakeholders to help advance product development. There's a high attendance rate. These are virtual meetings, so an attendance rate of 63% is high. And we haven't really done, I'd say that the attendance is high both from word of mouth, stakeholder outreach, and listservs, but it is very much attended. Some of the topics I think are quite interesting. One, it started out as a basic FDA CBER workshop for patient advocates, and then there was a seminar on Regenerative Medicine 101 just to explain what these various therapies were. A webinar on the critical role of patients in advancing gene therapy treatments for rare diseases. And there's some other topics also I'm not going to list, but one that I found really interesting was the April workshop was on clinical trials, the patient experience, where we recruited patients or family members to describe their experiences with participating in clinical trials. I think that's important, honestly, not just for patients, but for developers too. I mean, I'm sure that they do have a good idea of what it is for patients, but it's I think, always appropriate to think about what it means to be in a clinical trial from a patient point of view. So that is the end of my talk, and I'll just stop here and take any questions along with any others. Thank you.
Thank you. We would open up the audience for questions at first, and then we've got a few, if there are people who want to come to the mic. One question I think that I'd like to ask the panel that came up on another discussion point is that there's rare disease obviously is an international landscape. What type of collaborations or things that are you doing globally to connect with other jurisdictions around rare disease drug development? Would anyone want to make comments to that?
I think I'll turn it over to Carrie Jo, who does a lot with their clusters. Go ahead.
I can start with that. So rare disease really involves a global community, and we are very well aware of that. And we have extremely close coordination with our colleagues through a number of mechanisms, one of which is the International Rare Disease Cluster. And that is a cluster that involves the FDA, the EMA, as well as Health Canada. And we meet regularly to discuss development programs, challenges in rare diseases, as well as exchange information about initiatives and regulatory innovation that is going on within our space. And so that is one of the ways in which we do that.
Sounds good answer.
Yeah, I
Yeah.
will also add that specifically because cell and gene therapy are sometimes have some additional challenges, there's specifically also a cell and gene therapy cluster. And we have a number of other kinds of international educational efforts or capacity building for regulation, which I think are perhaps probably shared with CDER.
Okay, thank you. The other, I guess, one of the other things that I'd like to add to this is that in addition to this room, there are a lot of impacted individuals that are on this call that can't directly ask questions. So one of the things I think that you all highlighted was the FDA's interest and support of engaging the patient community. And I wanted to know if perhaps you could walk through what that experience or give some examples more concretely than what we're giving on the slide so that people will know what those meetings look like, what types of things do you talk about, what are the venues, so that as they're thinking through their own situation, they may be able to extrapolate, what that dialogue might look like rather than just starting from scratch.
So I don't know if you want, so there's both patient-focused drug development meetings and also what I
Patient
listening sessions, so slightly different. I think some of the patient listening sessions are really driven by the patients to tell us a lot about their experience and with the disease and what's important to them, which is incredibly, I think, reviewers get a lot out of hearing directly and really understanding. I don't know if you want to talk a little bit about patient-focused drug development, how that might be a little bit different.
I can, and I think what's critically important, a lot of these rare conditions, they can be really heterogeneic, right, in terms of the clinical manifestations, in terms of the experience of the patients. And so what gets discussed at, per se, a patient-focused drug development meeting is where the FDA review staff and communication staff really comes and is able to engage with patients representing a specific condition about which aspects of their condition are the most impactful to them. What is their unmet need? How do they feel about various aspects of how to best participate in clinical trials? And the FDA staff really listens to that, and it makes a difference when we go back and review a trial or help engage with drug development companies throughout the process of developing a pivotal trial, that we've heard from patient groups and can consider what is really going to be the most impactful from their perspective.
I can add to this. So with the two different types of meetings, the patient-focused drug development and the patient listening session, emphasize that both of these are patient-run events. So the patients do the speaking, they drive the agenda. FDA attends but is largely in listening mode. It's not there for a back and forth on regulatory issues. It's really for us to hear and absorb the patient experience. The patient-focused drug development meetings and the patient listening sessions are quite complementary. They are different, but they can complement each other. The patient-focused drug development meetings are usually organized events at a site. Depending on where it is, FDA may or may not be able to attend in person, but we at least try to attend virtually if it's not in a local area. But there's often panels of patients, preset questions, Q&As of surveys and things like that. So it's a little more formal in its structure, and it's usually a day-long event. And we do listen to those, and then a patient voice summary is written up out of that. But a patient listening session is something that's directed to the agency directly and happens on agency time. It's usually, lately since the pandemic, it's been virtual, although prior to the pandemic we had done a few inpatient. And it's usually a smaller group of patients who are expressly describing their lived experience with the disease. And so there's often 5 or 6 participants, and they tell their stories. And then at the end, the FDA may ask some questions just about specific symptoms or things like that. But again, it's not a forum for regulatory advice. It's really a forum for us to listen and hear. But I find them invaluable. I know our staff does as well.
No, thank you. I think the other question, I think, we've discussed quite a bit, and I think it was loaded with the accelerated approval pathway. There's a lot of supplementary tools around supportive evidence, and I don't want to go in great detail today, but if there are any pearls that you all want to convey around this, because there's been a lot of, I think, a little bit misconceptions about biomarkers, Michelle's favorite term, digital biomarkers, and then the use of real-world data, supportive evidence, and are there any initial pearls that you might want to share with the audience around how you consider those in the review process?
Yeah, I think my general advice is to think about this very early in the product development pathway, that surrogate endpoints are sort of a holy grail, but can be hard to develop and validate. But you can use biomarkers throughout development for various uses, even not necessarily in support of regulatory use, but for drug development in the space of proof of concept and early discovery. But later they can be used in drug development to help define the population, to enrich the population, to measure response, and then possibly used as a surrogate endpoint. But the confirmatory evidence piece of this is
Different.
more new to the agency. It's not new, it's been in the regs for a while, but we've put more emphasis on it lately because we understand that in a rare disease space, it's really unusual where you're going to have a single study alone that's going to be robust enough because you've got a small patient population. You're not going to have a large multi-center trial, and that 2 studies may not be possible. So the confirmatory evidence is a way of using some regulatory flexibility to try to substantiate that primary study. And what often happens, and I'll use the tofersen NFL as an example, is that NFL and tofersen wasn't their initial endpoint, but they knew they were going to measure it. And so it's important to have good quality data and assays very early in the design of your studies because you never know what could end up being used as a confirmatory evidence or even a surrogate endpoint in that situation. So if you have promising biomarkers, do the work early up front to make sure that you have good quality assays because you may find that you need to leverage them later for confirmatory evidence or potentially a surrogate.
That's good. Thank you.
Thank you very much for this fascinating presentation and sharing these case studies. I'm Cécile Olivier, I'm the Managing Director for CPAS office in Europe. And particularly in rare disease, as you know, there is a global context that needs to be taken into account. And when you have this situation with challenges around finding the regulatory flexibility, I wonder if you think that there is still an opportunity to develop tools or DDTs that are globally compatible, or at regional level there are with disease symptoms or standard of care differences that would require to focus rather on having DDTs that are focused on a specific region. Thank you.
Yeah, I guess I would say I'm not I'm not aware of what it would really preclude drug development tools from being employed globally. And so I think that we have a lot of conversations with our colleagues across the pond, as they call it, and beyond the EMA, because we are very acutely aware that these are small populations and you don't have the chance for multiple trials over multiple times. So I think that we are happy to leverage any of the work that is being done, within the FDA. We do have a drug development tool program that hopefully many find helpful, and we share that information with our colleagues.
Okay. Last question, I think, for the group. One of the things that's impressed me as I've joined CPATH and end of last year was FDA's commitment to working with the public. There's these individuals are reserved right now, but they clearly on calls have a lot of passion and a lot of interest in the community. And so I wanted to end this session with a bit of a hope. So what things excite you about working in rare disease and get you out of bed and charged to this, and what do you want to convey to the public about that?
Well, I'll start. I think, why we do this is because we see there are significant unmet needs, and, trying to get a product to a population that is going to really change or improve their life. I mean, obviously, we want to make big changes. We're willing to work with small changes as we work there. We want to make this more efficient. But we also, we are the regulators. We balance that because it's just important that we don't put something out there that really doesn't work and an accelerated approval. If you don't get that confirmatory evidence, we don't know. So it's trying to do the right thing so that we can move this field, so that we can get cures, and we can, really work together.
And I would just add from my perspective, I'm a pediatric gastroenterologist. These were my patients. This is what gets me out of bed in the morning. I'm an evolutionary biologist. I believe in moving things forward, always. And so I think the rare disease space for me is just a wonderful way to combine those things, do something that matters. It's a space that is ripe and is necessary for innovation. And I get very excited every day to go to work and try and change things for those that really need it.
Yeah, I can only echo those comments. And, obviously the patients come first, and most of these rare diseases are, devastating diseases that, really impact patients, their families. So striving to find treatments for those patients are always at the first, forethought. But, and aside from that, I find the rare disease space to just be challenging, but in a very fascinating way. I love the ability to try to innovate, to try to improve on existing processes, and to try to make trial designs more efficient, to utilize all of the data that's available to us. I just find it endlessly interesting.
I don't really have anything to add, except we look forward to continuing to work with patients and developers and other stakeholders to advance these therapies.
Well, if there's any other questions, we'll close out the session, and thank everyone for speaking today. And we'll have the next group come up. We're still getting people mic'd up, so we'll just be a minute. Okay, I think that's everybody. I think we can get started. I wanted to have a session. So this session will be focusing sort of on industry's perspective on relating to drug development. We've got some very talented individuals on this panel who are just, besides being extremely passionate, have really worked through some success stories. And I wanted them to be able to share some of their thoughts and perspectives around rare disease drug development. What I'm going to have for this session to open us up is each of you introduce yourself and maybe a little perspective about, what products you're working on and some of the areas that your groups are studying currently, and then we'll work through some advice and feedback steps. So, Angela, I'll let you start out.
Thank you, Colin. So I'm Angela Genge. I am the Chief Medical Officer for Curalis. I actually wear two hats. I am an ALS clinical trialist, drug developer for 20, 30 years now, and I'm based in Montreal at McGill University, and primarily have been driven by the patient's need in ALS because patients die in ALS, right? So it's essentially the equivalent of pancreatic cancer. So at Curalis, we are really trying to take that cancer model and introduce it into neuroscience. We're tremendously grateful for the Tofersen development program and have really focused our energies both in developing the molecules. We're in ASOs primarily, and are in clinic now with a Stathman ASO in ALS, but really focusing on a robust biomarker platform that we're using to really demonstrate target engagement and effect on disease mechanisms, in addition to improving our clinical trial outcome measures in ALS, which is a big side project of mine.
Gordon?
Thank you, and thanks for having me on this panel. I'm Kodrin Lungu. I'm at Pfizer, Global Clinical Lead for Rare Neurology. At the moment, I work primarily on our gene therapy for Duchenne program. I'm a neurologist by background, spent most of my career before Pfizer at the NIH, both intramural and extramural, so I'm used to, I looked at this first with the researcher eye, then with the funder eye, and now with the developer eye. So.
Hi, my name is Joanne Donovan. I appreciate the opportunity to speak with you here today. I've been in biotech for over 20 years, first at Genzyme, another biotech, and now a chief medical officer at Edgewise. For the last 10 years, I've been focused on Duchenne and Becker muscular dystrophy, and at Edgewise, we're developing a drug that targets the fundamental issue when dystrophin is lacking. That is that there is continued contraction-induced injury that leads to this devastating disease. It has been incredibly important for us to hear the patient voice, so I'm delighted to be here at this conference trying to bring new approaches forward for this incredible medical need.
Good morning, everybody. My name is Tom Miller. Thank you very much for the opportunity, Colin. I'm really glad to be here today for such an important topic. I work at a company called Rare Pharmaceuticals, and I am responsible for our acute, chronic, and pediatric care nucleus in our clinical development and operations function. I suspect most relevantly for the focus of this panel, the core area of portfolio responsibility that I have quite a bit of focus on is pediatric disease drug development. And I've been spending quite a bit of time as of late working with our satellite companies that are focused on development of regenerative medicines. Thanks.
Hi everyone, my name is Lauren Boak. I work at the pharmaceutical company Roche. I've been there for 16 years. I've been very fortunate for the last 6 years to be working very closely with the HD community. I lead the tominersen program in HD, which is an example which I think a lot of people have, in the room have experienced. There's a 3 steps forward, 2 steps back. Rare disease drug development isn't easy, but we're all here to work together and see how we can make it a success for all the diseases, and really, really pleased to be here today.
Well, I think to start the session, I think just sort of segueing from Lauren's talk, your comment is, like, do you have any thoughts? I mean, as working in rare disease, obviously it's extremely different than big pipeline drugs. I mean, we all know the challenges, and I was wanting to see if each of you would convey just things that you felt there was a success in thinking, and then a whoopsie, we probably should have done it differently as an example for your programs.
Maybe I can just kick us off because I started the segue. So we have been working in Huntington's for over nearly 13 years actually when we partnered with Ionis. But when we really at Roche started leading the development was about 7 years ago. And it was really important for us as we moved more directly into this space is to understand the disease, because we didn't really have that solid understanding, and what was really beneficial was the availability of real-world data. This is to understand the natural history. I mean, everyone in the room understands this, but even to understand what kind of clinical trials, what kind of endpoints we could expect to be able to use in our clinical trials. And we're very fortunate in HD to have access to a very rich set of data from the Enroll platform, which CHDI Foundation, we have representatives here, but also others in the HD community really came together to build. And I think everyone can agree that every single data point that is collected from a rare disease individual or their families is precious. And so the ability to be able to use, all have the access and ability to use all this data for all our purposes is so critical. I mean, this data set that we had really allowed us to hit the ground running with our clinical trial design and get started in HD. Obviously, there's still lots to learn and lots more to work together on. But I think that's just, having that foundation of data and information to sort of to start building, as we often referred to it, building the plane while we fly it. So that was really critical. But I'm sure there's lots of other experiences on the panel too.
Yeah, maybe happy to go next, as I've had an opportunity to work with CPATH and Colin and others for some time, and I often reference the fact that I would say most of my learnings, independent of formal education, has come from the School of Hard Knocks and learning what not to do. The rare disease development is very challenging, and this is my 27th year in industry, primarily focused on pediatric and rare disease, as I mentioned, and I noticed a pattern along the way with companies that I've worked for, that they tend to approach it in a fairly formulaic way, not too dissimilar from large pipeline products, which is a mistake. I think really trying to think differently about how you access patients, use of data sets, and I think regulatory science advancements and how we start to think about use of real-world evidence in our clinical programs is critically important. And from the last speakers from FDA, the reference to working with advocacy groups, in my mind, is a absolute critical success factor. If the advocacy groups don't understand at least the rationale, the fundamental rationale for your trial, good luck enrolling it. So just a couple of examples of really areas where I think are different than traditional drug development, large multi-center trials, and really encourage folks to really try to think differently as they approach rare disease development.
Joan, did you want to go next?
Yes. I would very much echo what you said about understanding the issues from advocacy organizations. And in Duchenne, one thing in particular that has been important is the advocacy organizations, PPMD in particular, stepping up with how do you sort out what the issues are in the endpoints that you're looking at. You know all of these things are important, but how do you look at even things like reproducibility and bringing companies, even before they're in the clinic, into that conversation in a non-competitive environment has been incredibly important. And CPATH is very similar in terms of bringing people together in a non-competitive, not just within the space, but beyond the space. So I think that is really important. I think one of the things, the lessons that I have learned is the importance, as was mentioned earlier, of a biomarker to identify that your drug is on target, it is hitting the mechanism that you are looking at. It is supportive evidence in terms of mechanistic proof of concept, but it is incredibly important in terms of selecting a dose. And because that is, I think, one of the Achilles heels in drug development in orphan, because we don't have the luxury of doing a 250 patient phase 2 study and choosing dose selection in multiples and then going on to a phase 3. We don't have the chance to do it over again. And that's in part because of the patients. It's also in part because many of the companies, yours excluded, are small companies that don't have a chance to keep doing it. So it's incredibly important to have that biomarker signal earlier, and that's been a lot of what we have also talked about in developing biomarkers with CPATH.
In terms of the positives and things that have worked, I think one of the things that has been great in this program has been the effort early on of translating the basic science and the mechanism of action into something that is functionally relevant. So there's been a lot of interaction with the patient community and with a lot of stakeholders and KOLs to learn that. And I think that's made the development successful up to this late stage. And I think that's that's again critical, and I think I'm echoing some of what the others have said. In terms of what could have been done differently, probably anticipating even more the complexity of a gene therapy in a rare disease and all the different things that you can't think of. And I don't know that anybody could have anticipated, just how complicated it gets in terms of safety and in terms of other aspects of development. Personally, since I come from a CNS space, I was used to gene therapy being delivered in the CNS, so it's much more comfy when you're hiding behind the blood-brain barrier than when you're outside of it. So, going back to your point about biomarkers and outcome measures, I think some of the environment that we just heard about now, in terms of the pilot programs and the FDA initiatives of developing outcome measures with sponsors, it would have been great to have this like 10 years ago when this program was starting.
So Curalis is a relatively young company, so we're learning from our mistakes, but more learning from the issues of others, right? But I'd really like to emphasize, which is where I think critical path really plays a role in ALS, is dogma. We have dogma handed down in ALS from the 1990s. The dogma includes, and it's really around trial design, and every time a young company comes to me, they come with something that was published 10 years ago, and this must be the way to do phase 1, phase 2, phase 3. And there are 2 or 3 areas where the dogmas hurt us in ALS. And one of it is our primary outcome measure. If you read a paper about ALS, it talks about an ALS functional rating scale that was developed and published in the 1990s. Now, most of us were born in the 1990s, but Coco Gauff wasn't born in the 1990s, so that's how old the ALS-FRS is. And it was developed for a particular reason, to be able to document the point and the clinical point in the disease that the ALS patient that you're seeing in front of you was when you saw them. It was transformed through a situation in which the other primary outcome measure was survival. And you can imagine that survival is not a very comforting primary outcome measure for ALS patients, because it implies that at least half of them have to die before you get to a positive trial. So hence the ALS-FRS became the primary outcome measure, is the primary outcome measure, and was in the tofersen trial the primary outcome measure that failed the drug. I have a lot of opinions about that program. I'm a big recruiter, and I feel very strongly that the FDA made the right call. So you can get me going for an hour on that one. But buried in there is that the ALS-FRS was never built to be a primary outcome measure. It has become one. They tacked on the respiratory component, but never validated it once the respiratory component was added. And then we implemented it across all clinical trials. What did we not do? We didn't look at how it was being given. We created these 2 well, actually there's 3 certification programs for the ALS-FRS as a primary outcome in ALS clinical trials. One is American, one is European, one is Japanese. There's no surprise in this room, right? Yeah. These three programs, until last summer, all trained their raters differently. Yeah. So, and in their training, they certified, recertified only once every 2 years. So you have the situation where the regulators and the small companies who don't have anyone who is buried in ALS, seeing this as the gold standard, there's no flaws in it, it's a perfect outcome measure. This is where CPath is going to help me a lot. It turns out that if you combine data from sites that are Canadian, sorry. American, European, and Japanese, you're actually combining data that is not the same. It's called the ALSFRS, but the raters score it differently.
Yeah.
If you change the rater, there's a huge internal bias. So you have an outcome measure that's determining the fate of drugs in a really important disease, in which you're combining data that has not been obtained in the same manner. So one of the big pushes last summer was that we harmonize the US and Europe so that ongoing trials will have one set of training for the ALSFRS. We haven't yet brought in Japan. They use a different formula. And we haven't really addressed the issue of the fact that staff turnover at sites. Now, this sounds really far away from the FDA, right, and regulators, but they only have, the data that they look at is only as good as how we've obtained it. So there is a real need in ALS to take, and this is an example of a primary outcome measure. If I was a Parkinson's doc, I'd be saying the same thing. And if I was in FTD, I'd be even more extreme. But we have a real need to make this outcome measure operationally more robust. And so for us, in the ALS broader community, the partnership with CPATH is going to be critical. We've actually discovered that patients are better than raters and site staff. And we're actually really actively looking at self-administered ALSFRS using an app, because you know what? It eliminates the bias of the raters and the site. So the only bias in the app is the patient's bias, which does not change over time. Whereas you change a rater, we had sites in the middle of a really important trial lose all of their staff over COVID, 100% turnover. So 100% of the patients had new raters. So you can tell I'm passionate about it. It's an example of how CPATH is going to help us. But the data quality should not be assumed, even if it's the primary outcome measure for 20 years. And we need to actually look at that.
Yeah. Tom or Laura, do you want to talk to endpoints and look at that?
Sorry.
The ALS is not an area
ALS, but,
that I'm particularly familiar with, but before you even got into the to the detail that you had referenced, immediately what was going through my head was thinking about the sensitivity, accuracy, and precision of a tool, and how reliable could it be if folks have to be trained on it, and so it makes perfect sense the direction that you have. To the degree that it's possible, I very much, for various areas that I focus on, I very much like to get a comfort level before we even think about picking an endpoint to understand how reliable that endpoint is in an appropriately translatable model, in a non-clinical model. I don't know if that's possible in an ALS-like setting, but for many of the areas that I work in, it is. And I like to get comfortable with biomarker X, Y, or Z in advance of even proposing something like that to a health authority in a non-clinical model. But I prefer as quantitative as possible.
So I'm curious on this, if a potential solution would be switching to a functional milestone-based outcome, particularly in something that moves as fast as ALS, as opposed to continuing to try to improve the scale, which is less than ideal to begin with. So
that's an excellent part of the process, which is looking at, we're not at a point where we can move away from the ALSFRS. We need to learn how to implement it operationally so the data quality is better across countries. But time to event, we're looking at different outcome measures that are much more common in oncology, say, but have a real relevance in ALS, it had real relevance in Huntington's, real relevance in FTD. So you can tell where else I've been. But the concept of pulling from not more established, but bigger areas like oncology, things like time to event or other outcome measures that you can more easily objectify and remove the operational aspects of the outcome measure.
The analog in Duchenne is the 6-minute walk.
Exactly.
12 years ago, the companies had different protocols for the 6-minute walk. So an assessor would use the wrong one. And so as opposed to now, there is basically a academic-based group that is training all the centers for all the companies. And it's a monopoly, but it's a good thing that everybody is getting the same training and getting it on a very consistent basis.
Maybe just I can add, it's obvious that there's so much that can be learnt across diseases. And in Huntington's, as you mentioned, Angela, this sort of time to event proposal is something that is worth considering. And while we do have some endpoints that do okay in the later stages of HD, we can deal with them, we can do trials. Really the impetus is to move into earlier stages of HD in a similar way in Alzheimer's as well. And we're fortunate to have an integrated staging system that's both based on biological and clinical data. You'll hear more about that over the course of these couple of days. But this is really where we can learn about like time to a functional change, like from stage 2 to stage 3 in HD, or then looking at biological changes, because the clinical measures that we have do not change in HD from the earlier stages and up until a very, very late. And with our increased understanding of the underlying biology and how perhaps intervening earlier may be needed, this is going to be really critical.
So Colin, I'm wondering, I know this was on your list of topics to discuss, if this would help us solve another problem that you wanted to talk about, which is outcome measures to use for long-term follow-up or post-approval studies, right? Because you run into another issue of are these outcomes translatable to the real world? And a lot of what we do in trials, particularly outcome measures that require intense training and uniformity in how they're administered. So.
Just one additional comment to that is that obviously you're working in gene therapies. How are you attacking the follow-up periods and like standardization of what kind of data collection and following up patients who may move in and out of healthcare systems?
Yeah, it's a headache. I'm on that topic. I'm here to learn more than to convey, because we're struggling with this, obviously, and I think the input we get from KOLs and and from patient communities is that, and I think I love your idea, right, of going to patient-reported outcomes, and I've learned over the years that in medicine amazing things happen if you listen to the patients. So.
Tom, you have
Yep. While this concept as it relates to regenerative medicines is new to us, it's not, the concept is not new, right? I mean, for pediatric rare disease studies, we have to do long-term follow-ups all the time. This is a typical thing. But one piece that's always fascinated me, and back to the headache topic, that I haven't been able to figure out yet is how to do better in the United States. I find that long-term follow-up is much more consistent, and patients are much more accessible outside of the United States. And I suspect it's because, and at least one of the drivers is we tend to change our medical homes a bit more here than folks in other countries. But this is a particular challenge that I would urge all of you that are in the development space to really think through strategically and tactically how you're going to keep track of these patients. It's hard.
Yeah, and it's particularly difficult in something like a gene therapy space where the administration of the therapy tends to happen in a big academic center that's all set up for this, but the follow-up won't.
Yes,
so
exactly.
Very different instruments available sometimes.
So we have actually proof that it can be done. And it's not based on a drug development program, and it's not based in the US, so I apologize. But in Germany, the ALS Clinical Trial Network has done two different things that I think, in fact, I've connected people to really learn from. And one is that they have developed an app and put it out there to all of the German sites. And it's in a mobile phone ALS-FRS-based app. And the patients in the clinics are using this app. I don't know how many data points, 10,000 data points or something they have over 10 years. And then the second thing they've done is they have set up a neurofilament assay program, and they now have several years, well, at least 2 years of data on all patients who are followed by the predominant ALS clinics in Germany. Their neurofilament levels every 6 months. So they have a huge data repository that they've built. But again, the issue of what country you're doing this in really comes up. Germany, people tend to stay in the clinics that they start with over time. Most of Europe is this way. The US is the opposite, even with a disease like ALS, particularly because what happens is people develop ALS wherever they're living. They often have to move to access their family resources in terms of care as the disease progresses. So even in a disease like this, you get a lot of movement across the US. But I think there's still opportunities as we connect bigger groups to work on this. And I think this is going to be critical because we'd really like to get away from placebo-controlled trials in this disease. And we can only get away from it once our observational data is more robust.
Okay. I mean, I know, Lauren, you spoke to this a bit earlier, and obviously CPATH is a big fan of data. How are you all using real-world data in your programs, and what things have you found useful for its use in various steps of rare disease drug development? Tom's a big fan, I know.
Yeah, I am. And we've had some recent success in this regard. We oftentimes have very challenging studies in pediatric settings. And in this particular example, this was a rare complication of prematurity. And it just so happens, and I tease my friends, I'll maintain anonymity of the other company, but they had done a very similar trial in the identical patient population about a year or so staged between us. And we looked at the control population from their data that they published in a very robust, high-impact, peer-reviewed journal. We spoke with our statistical colleagues internally about alternative statistical methodologies, in this case, a Bayesian-based approach. And we proposed this approach to multiple health authorities around the globe. And we effectively were able to borrow real-world evidence from a very well-conducted trial with, I'm sure, outstanding data quality, as is typical in our industry. And we're now registering this indication around the globe. So this is not going to be a solution for every trial that we pursue, and I guess we got a little bit lucky with having this cadence and pacing of the two studies. But what I also found, which was, I guess in retrospect, shouldn't have been a surprise, but when it was time to consent, the caretaker or mom and dad, our relative ratio of success was much higher than in our other studies. So our operational throughput for the study, the actual speed at which we could conduct the study, improved dramatically relative to traditional control methodologies. So again, this is not going to be a solution for any and everything, but in certain circumstances, I appreciate health authorities' openness to at least engage in the conversation. We still should be held to the same tier one level data standards that should accompany any trial. But in certain circumstances, this can work, and now we have some real proof that it can work.
Maybe I can add sort of more of the cautionary tale. Obviously, this is a wonderful example. And just maybe reiterating the importance of quantitative modeling and learning from as much data as possible. As mentioned earlier, that we have this wonderful registry of data in Huntington's on the Enroll platform, and which tracks the natural progression of disease. More in the later stages, there's still some gaps in the earlier stages of disease, but we can learn a lot from it. The phase 3 study from tominersen, and tominersen is an ASO targeting the Huntington, the underlying cause of HT. There was actually quite a lot of placebo effect, well, quote unquote placebo effect. And I mean, there's a lot of things that could have contributed to this. There was a lot of hope and expectation behind this trial. Then, it's a 2-year trial, so, perhaps placebo effect over time would start to diminish. But it's really just a reflection of how important it is to learn from not just natural history observational data, but also what happens in a clinical trial setting, and try to adjust for these factors. There may be factors around the azos. It's obviously intrathecally injected, and so there's some aspect of the actual modality and how it's administered that needs to be factored in versus an oral, etc. But all of these things are really important. Just reiterating the work that in the quantitative modelling groups at Critical Path do try to integrate, or even internal companies work on, to integrate these different factors. Because I think a few people have mentioned it, we'd love to be able to go away from placebo-controlled trials ultimately and be able to have these new ways of creating like digital twins, all of this technology that is becoming more and more available to us, but there still needs to be a foundation of data underpinning that. And so getting the right amount of data and the right amount of knowledge before we sort of embark on that is critical, and we can only do that when working together.
Anyone else have any thoughts?
So the ALS community has recognized one other data set that we need to figure out how to get access to, and that is the failed clinical trials. So there are trials that don't meet their primary endpoint due to trial design, but there are a number of clinical trials over the last decade in ALS where the trial simply failed. There's no difference in the lines, no matter what way you sliced it. That's an incredibly valuable data set. That data set that is essentially a full placebo data set, unfortunately. And some of these data sets actually have neurofilament data in them, in ALS. And so you've got these big pools of, I can name 4 different relatively large trials for ALS. It doesn't compare to stroke, but it does, it's big for ALS, where that data could just help the entire community. And if we could all have, if there could be a way to access it in a de-identified manner, where we could use that for our modeling, we could use that for our predictions, it would help us with a number of aspects of our clinical trial design. I think that's it's not real world evidence in the traditional sense, but it's a lot of evidence.
Yeah, we've done that in Duchenne as well. That's there are trials for. Yeah.
And just related to CPAT in particular, the consortia that I'm involved with, we do have a data lake amassing exercise underway as we speak. So this is an approach that is not unfamiliar to CPAT.
Can I just add one more thing? I'm sorry, I feel like I'm the cautionary tale telling. It's more just like a sort of a comment rather than, a question or a comment, like how, when we're looking at old data, particularly biomarker data from older studies, how are we sure and confident in the assay and the technical validation and the confidence in the data? I think that's a really important thing that we all need to be very mindful of. I think assays, NFL assays and other assays have really made lots of progress now, but that's now. And so we just need to be to work together on how we can make sure that we can be confident. I think Dr. Baracchio actually mentioned it in one of her comments that we need to start early to make sure that we do have that technical validation and confidence in the data, because garbage in, garbage out.
Yeah. Indeed.
I can add to that a little bit of plug. I think RDCA-DAP is well suited to that, and I think that will be sort of on our agenda. But in addition to having the experts and the stakeholders at that and reviewing the data and trying to dissect it more thoroughly, with the QuantMed programs that we have, it's just a perfect marriage. One of the reasons I joined CPATH was just the ability to do these type of things.
Yeah, you had mentioned the placebo effect, and it's by aggregating this data. DRISC is looking to aggregate this data. CTAP has aggregated the data and looked at the question of whether or not the placebos are different than the natural history. And it turns out that they aren't in that disease. And it seems as though that is an effort-based measure, but at the same time, you see that the placebos, when matched, are very similar to, or basically identical to the natural history. So it goes to the how can we minimize the placebo in these trials. But knowing that information, that it doesn't differ from natural history, is critical to being able to go forward there.
And on the topic of natural history studies, this has been a new area of interest of mine in particular, because sometimes the real world evidence that you need or you might like to rely on simply doesn't exist. So sometimes the best real world evidence that can be created is what you create yourself. So we've been thinking about gating natural history studies for some of these rare disease programs for a number of reasons. Number one, to create the evidence that we could quality control and make sure that it's robust enough, unlike many registries, for example. But we're also getting, it's almost like an operational pre-feasibility, so you could get a sense of how the centers are actually able to access patients for you and who you could rely upon when it's time for the live game. So there's real value in thinking a bit differently about this type of approach and in these kind of circumstances.
I'll open up the floor to questions. I've hogged the question space, but if there's others that want to ask questions, I see Susie McCune getting up. I'll let her, others.
Hello, I'm Ahmed Pindon, Chief Scientific Officer, CP Disease. We are a particular disease because we have one mechanistic, the VCP, but the problem is that we have patients in ALS, in Charcot-Marie-Tooth, in myopathy, Parkinson's disease. So on the first part, preclinical, we have one target, one mechanisms. But then after, when it comes to outcome measures, to how can we do that and integrate all these different manifestations, how can we, in drug development, think about outcome measures that can be across all those diseases. And I think we are not the only disease with this problem. And if you take the rare disease from ALS point of view, for example, you have to choose which one of all the genes you are going to choose. So if you take the other approach of centering on a mechanism, but then after, I think it's a headache for all of us, after that, in clinical phase, how can you make the outcome measures?
So you talk about phenotypic divergence from the mechanism, right? And how you
single mechanism, but then you have from ALS to Parkinson to Charcot-Marie-Tooth.
And there are no biomarkers that reflect pharmacologic engagement downstream that would be common to all these phenotypes?
Well, that's what we are looking for. We are looking into NFL, we are looking into scan, but on the really measuring the outcomes, that's our headache, and I think it's something we should think about because we approach rare disease by ALS or Charcot-Marie-Tooth, but in fact it's not one disease, it's a lot of diseases.
There are attempts now, right? And the Wave Life Sciences did a basket trial with their C9 targeted therapy. So there are natural partners and disease partners for basket trials. Some are very, very different, and NFL was a good comment because, but they successfully ran a study with ALS and FTD in the same SAD-MAD study. The study is closed, but the ability to run, they demonstrated you could do that. But each of the arms had the outcome measures appropriate for that disease. What was common was the biomarkers. So I think that's in situations where you have a molecule that is targeting a pathway that is common across, say, neurodegeneration, I think that's a very doable thing as we get more robust biomarkers. And that's going to be the essence. And then once you get through your SAD-MAD into POC and you're looking at your pivotal, that's when you split off. But your early data will be driven by biomarkers, in my opinion.
I like that.
Thank you.
Susie? You're like...
Susie goes first.
Age before beauty, right?
Wisdom.
I'm Susie McCune. I'm at the clinical research organization called PPD. And just wanted to remind everyone, I'm in the pediatric space with Tom, and the Duchenne space as well. About 50%, if not a little bit more, of rare diseases are pediatric. A lot of them are congenital. And so we see manifestations from infancy through childhood, through adolescence or toddler, childhood, adolescence. And the outcome measures change over the course of each of those periods. The way we capture them change, especially quality of life questionnaires, especially for the patients and the caregivers. And as they get into adolescence, their approach to things may be very different from their parents. And so the 6-minute walk test being one that reminded me not only of not being able to do it when you're really young, but also I heard stories of parents literally grabbing their child's hand and dragging them down, the runway. Right, so just a reminder that that's a complication in pediatrics as well, as we struggle with outcome measures, period, but then add on to it that they will differ through the course of the life of a pediatric patient.
Yeah, and we do, and I suspect many of my colleagues that do pediatric studies often strive to go as low in the age range as benefit risk will support. So, but this is the inevitable challenge that we have, right? What measure of exertion is appropriate for an infant versus a child versus a teenager?
And even the quality of life questionnaires, have they been validated?
Exactly.
Yeah, and
Preaching to the choir, guys. Anytime I can say neonates, and certainly with congenital rare diseases, we do see the manifestations. Many of them, especially the severe neurodevelopmental manifestations in patients in the NICU that can't get off the ventilator, and so looking at outcome measures for those patients that aren't just survival, as was mentioned earlier, but also functional outcomes.
Thank you, Susie. Good to see you.
Yeah, so Klaus Romer with CPATH. I just want to introduce one clarification and then a question. Time to event is really a data analytics approach that can be used to define the primary analysis of a trial, not necessarily an endpoint per se. But, Angela, thank you for bringing up the fact that we need to stop what a friend of mine calls the last paper carried forward approach.
Yes. Yes.
We definitely need to stop that. But thinking about the longitudinal component, how important it is, that's one of the beauties of the time to event, time varying probability and all that, looking at the entire movie throughout the trials. Speaking about that, can the panel comment a little bit on you guys' take on the use of digital health technologies in these kinds of trials for rare diseases?
Who wants to start?
I can start.
We all have an opinion.
Yeah, I think digital health technologies are very, very promising, and because clearly the amount of data and that you can collect outside of clinical visits it provides more sensitive evaluation of effects over time. I think we still need to be very conscious of the additional burden this takes in clinical trials as well, and it's always about making sure that everything that you collect is going to be useful and used used for a purpose, because it's time and burden on individuals and their families during the clinical trials. But overall, our experience, we've got experience in Duchenne's, but also in Huntington's and Parkinson's of developing digital monitoring tools, and it's really at this point in time very much complementary to what's existing, but I'd love to see a future where these will be the tools that are only needed, because they particularly may be more sensitive early on in stages of diseases. And it's just adding to the tool armatorium that we need for our drug development programs.
Mm-hmm. It's kind of natural in ALS, because ALS is a movement-based disorder, right? You lose various movements, so it is definitely, there's a lot of interest. The simpler, the better. Digital health, they like really complex things, right? And that gives you a lot of data. And I like simpler things because I want cleaner data. But I think it's a huge step forward, and it actually applies to a number of neurodegenerative diseases, Huntington's, ALS, but PSP, certain variants of FTD. It's actually going to be a huge tool.
There's actually been a very successful track record of developing it in Parkinson's, and it's moving in that direction a lot. So I think it's the future. And one tidbit that I've learned actually trying to develop an app for Parkinson's is that adaptation, the take on of technology and the use and the compliance with it vary a lot, and it tends to go up if the patients get some benefit or some use out of it, as opposed to just being a chore and wearing it so the doctor is happy. So we tried to develop an app for Parkinson's. Nobody was using it, and when we made it dual function to remind patients to take their medications, then everybody was using it. So I think it's something to keep in mind.
The same in Duchenne, that if you're looking at a muscle disease and you're looking at function, you're looking at how fast the kids run every day and what their maximum speed is, or they're close to maximum speed, that's clearly speaking to feel and function. Absolutely. And you see it in real world evidence, as opposed to the 5 seconds to go upstairs in a clinical trial setting.
And then I think this is especially true in studies or patient populations that have a pediatric component as well, because if you could measure something without requiring an office visit or another stop, you have to remember it's not just the patient in your trial, it's the caregivers that have to get them there, it's missed work, missed school, missed afternoon sports, so forth and so on. So trying to measure as much as we can reliably and robustly without requiring a car trip or a plane trip, I think benefits everyone.
I just wanted to make a comment. We seem to have had a really lengthy conversation on endpoints, and I think it's important to remember a couple things with some of the questions that have come up. So first and foremost is, what is that patient experience with that disease? Because even in families of diseases or genetic pieces that which disease will appear, the example with FTD and ALS, those patients' individual experience is going to be very different, and what they really think about their outcomes and their progression of their disease is going to be very different. And so I think we need to not forget about that when we're thinking about the family concepts or the mechanistic concepts, is that in the end, how a patient feels about their disease and their experience is going to be very unique and needs to be critically considered when selecting an optimal outcome to support a clinical trial endpoint. So I think we need to remember that, because while mechanistically it looks great, it may not actually be the right thing to measure in the end, which ties to the other pieces. What do we think our drug's going to treat or our biologic's going to treat? And that's the piece that I think a lot of folks sometimes forget about when they're selecting outcomes and endpoints, right? We're really good at taking in the patient voice, and we've come a long way in that way. But we also have to make sure we know that what we think the treatment's going to do, we're measuring that. So we're not questioning when we get the data that comes in and reviewing it, going, well, how far off was I when I captured? And so that's why we encourage early and often engagement with the agency. CDER, CBER, come talk to us. We want to help optimize those trial protocols and endpoint selection when possible. But we can only do that unless you come talk to us. And so I think that's a really important conversation when we're talking about our rare disease clinical trial development, because we don't have a lot of opportunities. And we definitely are on the same side of, we don't want to waste patients. We don't want to waste, we only have a small amount of patients to be able to recruit from. So I just want to remind everyone to do come and talk to us early and often.
Last question,
Great,
Susan.
thank you.
Susan Ward, CTAP. I thought this was a wonderful discussion of endpoints and all the pitfalls and ways of trying to avoid them, and ditto around use of real-world data. But there's one piece that I think I'd really like to hear people speak to that hasn't been raised, and that is accounting for heterogeneity. And, you know, we talk about heterogeneity a lot, and sometimes that's different symptoms for different patients. But I think Duchenne is an interesting example, because for each individual patient, the journey is remarkably consistent. What's different between patients, I mean, in the scheme of things, and patients may not say that, but from an observational clinical perspective, there's tremendous consistency, except for the rate at which the disease progresses. And so the heterogeneity is not so much around the symptoms, it's around the rate at which patients move through it. And so the question I want to raise is that there are ways in which one can, obviously that leads to a lot of variance in trials. And obviously you want to overcome that, but you can have perfection in all your endpoints. You're not going to get away from the fact that that disease is moving at different rates in different patients. So to what extent do you guys rely upon or utilize even statistical approaches that you can use in analysis, not design, but in analysis to reduce the variance?
So I'll make a quick comment and then hand it down because I think others have more. In ALS, we have a problem with statistical analysis.
Yeah.
There are at least 12 different statistical approaches to the ALS-FRS. So one of my requests that Colin and Elsa are receiving are actually to bring, there are brilliant statisticians that have been involved in ALS trials over 20 years, and yet there's still 12 different ways of analyzing it. And then when the company puts it to the FDA has to reanalyze it because they're all using different. So one of the things that we have to do is make the statistical analysis plans more homogeneous, right? And then within those plans, we need to stratify and follow. We do have some, in ALS, people talk about heterogeneity, but it is a disease of motor neuron loss. So there is a course, and people follow that course, and they're either fast, normal, or slow progressors, even though the milestones may look different along the way. So we now have a number of algorithms that have been developed that can predict whether or not it's a fast, normal, or slow progressor. So that's starting to be incorporated, but it's at the beginning.
And I think the dilemma that we have as it relates to prioritizing a purely statistical strategy is the sample size that you're starting with in the first place. These tend to be relatively modest sample size studies, particularly in the ultra-rare diseases, right? So your ability to meaningfully stratify based upon a statistical strategy alone is going to be tough. I prefer more of an operational approach. The concept of basket trial came up earlier. If we're able to endotype the disease in early development, if we know that there are different courses of disease progression, for example, and then test your treatment effect early on, if there is one, and then prioritize that patient population for your subsequent late development programs. So I prefer more of an operational approach to homogenize than a statistical one.
Yeah, I mean, what we've done in Duchenne is a bit of an all of the above, and so I don't think you can decouple the analysis from the design. I think you need to go in with keeping that in mind. And for anyone who doesn't know, so Duchenne, everybody follows this course where they improve just by natural development, and then around age 7 they start going down. And there's very few predictors. There are some predictors that can maybe suggest that the kid is going to go faster or slower, but they're not great. So I don't know, other than following the advice from one of my mentors of making friends with a good statistician, I don't have a solution.
Yeah. I would say, though, even though they're not great, they improve things.
Yeah.
And so just
Sure.
if you can improve the signal to noise,
Right.
you can improve the power consistently. So by adjusting for baseline function, as well as potentially as we learn more, these are long trials, so we learn more, we may end up analyzing it with additional information about what study population within that group. And you may not have to define it operationally, but you may be able to do your primary endpoint on a smaller group that's more homogeneous with additional information.
Yep, agree.
I'm just building on what everyone has already said, really. It's the enrichment at the beginning of the study is really important, plus also the pre-specified analyses of subgroups. And I mean, Huntington's similar to Duchenne's. We understand the underlying cause, but there's still a lot of variability. there's a mutation, but then there's different CAG repeats, and the number of CAG repeats only accounts for 50% of the variability. And so the worst possible thing is that you do try to study and then it fails because you haven't predicted your placebo decline appropriately. And so trying to understand what factors that you can adjust for at the beginning. Algorithms, as you mentioned, Angela, we have prognostic indexes that are integrating different data points. We can only get better though, because the more that we can at least understand what the trajectory, if we're using placebo arms, or to design our trials at the beginning. Because as I mentioned already, the last thing you want to do is it to be a clinical trial design error, the reason a trial fails. It needs to be, no trial is a failure. Obviously, we learn everything, and learn a lot, even if it doesn't meet what we were expecting. But we can try and do as much up front, but also at the statistical analysis plan stage as well.
Well, thank you, everyone. We'll close this session. We appreciate the expertise of this group, and we look forward to the conversation.
Hey, everyone. We're going to take about a 15-minute break. There's some refreshments, coffee, water outside. We're a little bit behind schedule, so we'll just give you about 15 minutes. We'll start back here about 10:15. So thank you. Okay, thanks everyone. Next I'd like to welcome Colin Hovengay back up. We're going to talk a little bit about the ALS landscape and some of the things that we're working on with Critical Path Institute and our partners.
Did you want this? Are you going to sit to speak? Okay. Everyone, I mean, this is an exciting time. This is one of the reasons I joined CPAth was, to work for the Act for ALS initiative. It was Klaus, I think door number 3 of our game show offer. And so it was an exciting opportunity to work with I've come to know some very incredible collaborators and individuals who are very passionate about working with rare neurodegenerative diseases. And so this session will focus on our efforts that are surrounding the Act for ALS. We'll start with Jacqueline, who you were introduced before, Emily from NIH, and then Alessio Travacchio for FNH. And they'll be discussing some of the efforts that are going on currently and all the great projects that are being planned in real time, as this is really moving very quickly. And I'll pass it to Jacqueline to open us up.
Great, thank you, and thank you for having me to speak about the Act for ALS and where we are today. So I'm going to focus a little bit more on FDA, but of course this is really a joint effort between FDA and NIH and our 2 public-private partnership partners. So as many of you know, just to level set, passed in December of 2021, it's Accelerating Access to Critical Therapies for ALS Act. We just usually refer it as the Act for ALS. And it had a couple different sections. Section 2 is really geared towards NIH. We won't spend a lot of time, but really using expanded access to do scientific research and use all the data. And then sections 3, 4, and 5 are really ones that FDA had to focus on in partnership with NIH for the public-private partnership, as well as an ALS and other rare neurodegenerative disease action plan, and then our own rare neurodegenerative disease grant program. And I'll spend a little time on that today. And then eventually there will be a report out from the GAO on what we've accomplished. So one of the things, as I said, we were to implement a public-private partnership, and we did that in September 22 when we engaged Critical Path Institute as a convener. And this has recently been expanded, as you'll hear about today, to include FNH and the new Accelerating Medicines Partnership into ALS. And so we're really, very excited about the synergies that both CPAth and FNIH bring to this effort. We were asked to publish and implement a 5-year action plan, which we did, and my slides will be available. That link is live. And included in that was an ALS scientific strategy. And then what I'll spend some time talking about is our award or grant program to public and private entities for research in this area. And just to give you an idea, what we were asked to do was really award grants that were about developing, diagnosing, mitigating, treating, and curing ALS and other rare neurodegenerative diseases in adults and children. And a pretty broad sort of mandate. It was to characterize those diseases in their natural history, to identify molecular targets of neurodegenerative diseases, and then to increase efficiency and productivity of clinical development, including the use of master protocols, add-on trial designs, and clinical trial networks. Now, I will say that, and you'll see in our grants, it's a little hard in a smaller grant program to start a new clinical trial network. So we've really tried to focus on what we think we can accomplish and get results on in the near term. So we stood up the Act for ALS task force internally, and one of the things we wanted to do was reach out to all of our SMEs within CBER, which is Center for Biologics, CDER, which is our drug center, CDRH, because this is across an FDA, so it also includes our device center, and NCTR. And we asked, where are the biggest opportunities for regulatory science? And what we heard was equivalency studies for clinical outcome assessments. Again, sort of going on that theme of can we make trials more decentralized and more accessible. Natural history studies, again, emphasizing the need to further understand these diseases. Our colleagues from CDRH really wanted to work on the enhancement of devices with brain-computer interfaces, and also to think about what about FDA cleared, approved in vitro diagnostic tests for use in diagnosis or treatment. And then, of course, basic translational research related to pathophysiology is always going to be a priority, something probably NIH will do more of than we will in our grant programs. So this was our FY22 awards. Again, we focused on, as we sort of prioritized, 3 different natural history studies, one in ALS, one in ataxia telangiectasia, and the third one in myotonic dystrophy type 1. So these are all 4-year studies, natural history studies. Then we did some contracts where we are a little bit more involved, and one of them was on the ALS functional rating scale, which I know we just had a discussion about, but really starting to think about remote use equivalency study, as well as a landscape analysis of computer interface-focused patient preference studies in ALS, and that was the first part, which was in landscape. Now, we did have an award, RFI for 23. We haven't announced the awards for this year. They'll be coming shortly, so stay tuned for that. I'm going to talk a little bit about the contracts and why we did this. So again, you heard about this in the previous talk, but we really wanted to look at whether we could get, to the extent that the ALS FRS may not be ideal, but it's certainly used, it can be then brought and used in a more decentralized way. Can we decrease burden, as you heard, improve efficiency, and think about greater diversity and inclusion? And this is just a description of the tool. I won't go over it. I think you've heard a little bit about that. And so, oops, back one, sorry. There we go. So the objectives is really to try and assess comparability of remote use scores compared to in-person scores and assess equivalency between a remote use tool administered via audio platform versus internet. So we have 4 tasks, 2 of which have been completed. One was the landscape analysis of remote use, and a manuscript will be coming out on what we found on that, as well as a pilot study of 12 semi-structured interviews, and that's been done, and that's to identify facilitators and barriers. And so the next part is really doing a feasibility and comparability study at 9 sites geographically dispersed in the US. And included in this is translation and cultural adaptation of the tool to Spanish. We want to do that to also make it more accessible. Importantly, as part of this contract, the Office of Administrator really wanted to make sure we had patient input into it as we develop this contract. So we have a patient committee with 3 people with ALS and 3 caregivers to provide ongoing feedback and make sure we're moving in the right direction. Sorry, I keep clicking too much here. The other part we wanted to do was the brain-computer interface, and this is our colleagues in the Center for Devices. And we wanted to see really what's out there, what's the literature about in terms of patient preference studies. Again, these are communication devices, computers, social media. What do we know about what patients think about them? What's their priorities in this area? When we did the literature and landscape analysis, there was not a lot of information, actually. There we go. We're a little sticky today on our... I don't know if you can get me the next slide. I seem to be... Thank you. So there was only really one BCI-focused study that we could find. It was a sample of locked-in patients as well as 13 patients with ALS and their care partners. So there was 3-hour home visits and multiple choice. It was an interview. Direct and private forms of communications were the most desired functions, and the only thing the study noted was a little bit of a non-alignment with patients and their caregivers, whereas patients preferred to wait until a BCI was needed, and their caregivers really preferred to have that information a little sooner. So we're going to move on from this, and actually we put out an RFA. So this is really to look at systemic review of clinical outcome assessments for communication brain-computer interface devices. So again, it's what we call this U2, U3 cooperative study, in which the first phase is a systemic landscape analysis, looking at the literature and looking at what's out there in clinical outcome assessments. And then the second phase will consist of patient and caregiver focus groups, collect information about symptoms, functional status, and perceived benefits and risks. So really thinking about how do we move the development of these BCIs forward. There are other, as I said, in 23 we have funding. We had about $5 million funding. What we did was we opened an RFA, an open docket, so that we could understand a little bit of priorities, and we tried to gear that RFA towards what we heard in the open docket. We had 21 comments. Again, improve the understanding of the pathophysiology and advanced biomarker development, things that are probably very familiar to all of you. We are now planning for FY24. We'll be utilizing, again, the concept ideas from stakeholders to develop a new RFA to address unmet needs for neurodegenerative diseases. I just want to also make sure everyone knows is that the Office of Orphan Product Development has a number of grant programs. So this is not limited to rare neurodegenerative diseases. It's their clinical studies of orphan products, and that RFA, the receipt date is in October, but it's every year. There's another one on natural history studies. So although we funded some natural history studies under the Rare Neurodegenerative Diseases, there's other opportunities to have funding for natural history studies. And as I said, the Rare Neurodegenerative Disease RFA for FY24 is in the works. So lots of research that we're trying to promote to inform our regulatory science. This is just contact information. Again, if you have any questions, we're happy to answer them. And I just wanted to really focus on the research and then turn it over to my colleagues in NIH and in our public-private partnerships to talk about the other activities we have. Thank you very much.
Thank you very much. I'm really glad to be invited to speak during this session. I'm just going to give you an update about NIH activities that are relevant to ALS research. So at the beginning, I just wanted to sort of give a brief synopsis about the disease. As you all know, ALS is a very, very challenging disease. It's an aggressive disease. It causes the rapid degeneration of motor neurons that control voluntary movement as well as breathing. And according to the National ALS Registry, there are at least 32,000 people in the US who live with ALS, and that translates into a lifetime risk of 1 in 400. ALS is characterized by significant clinical and genetic heterogeneity, which of course adds to the challenge in finding treatments. And only about 15% of ALS cases have a family history, which means that the great majority of patients get the disease sporadically, and we really don't understand the causes why these individuals develop ALS. And there are some promising news. By now, we have 4 FDA-approved disease-modifying treatments for ALS. These drugs may have beneficial effects on the disease progression. They may also extend survival. But it's really important to remember, so far there is no known drug that either stops the disease or reverses ALS. So there's a really huge unmet need. And the figure on the right, that is a graphic that I took from a review article from Pamela Shaw's lab at the University of Sheffield. And I think it really showcases very nicely the biological complexity of ALS. So over the years, many genes have been identified that either cause ALS or are risk factors for the disease. And these gene variants map onto different pathogenic cellular pathways that are either relevant to motor neurons or glial cells, and sometimes also interneurons. And these discoveries are opening many avenues for therapy development in ALS. And I think what is really positive in the field that there's a lot of momentum in ALS research. So currently there are a number of efforts underway to develop gene-targeting treatments for the familial forms of ALS. And in fact, earlier this year, we had an approval by the FDA of a drug called Qalsody, and that is for SOD1-linked ALS. It's relatively rare, but it's an important form of the disease. And parallel efforts are currently underway to also develop gene-targeting strategies for other causative gene variants, including FAS, also C9orf72 repeat expansions, and many more. But what I really wanted to emphasize that there are also new therapeutic targets emerging for the sporadic form of the disease. And these molecules are not only of high interest as therapeutic targets, but also for biomarker development. And I think we heard that in earlier conversations today, how important biomarkers are to optimize drug development in clinical trials in a disease that is as heterogeneous as ALS. This is really particularly important. So last year, NIH led a strategic planning effort for ALS, and we did this through a working group process. And all of these working groups included people with lived experiences in ALS, as well as scientists and clinicians and other subject matter experts. And we had formed 5 working groups that addressed different domains of ALS research. And each of these working groups developed recommendations, which we then pulled together in one strategic plan. And this plan was approved by our advisory council at NIH, or specifically at NINDS, in early February 2023. So we are now in the early implementation phase of our strategic priorities for ALS research. And in fact, on November 6th, NINDS is going to host a community update in which we sort of are going to report out to the community about our efforts to implement these ALS strategic priorities. And we're also going to hear from our nonprofit partners in terms of their recent efforts aimed at advancing ALS research. And the event is going to be fully virtual, so I hope that many of you will be able to tune in. And during this event, we're also going to talk about our efforts to implement the Act for ALS legislation. And of course, this is something that we're doing hand in hand with our partners at the FDA. So I don't think I have to sort of talk a lot about it. So Jacqueline already covered it, but certainly I think the passing of the Act for ALS legislation was a real milestone in the field. And the legislation has multiple provisions, and 2 of them are directly relevant to NIH. And one of them is Section 2, which calls for the development of a grant program for research on expanded access studies in ALS. And the other one is Section 3, and that is the establishment of a public-private partnership in the ALS space, and that is something that we're going to do with the FDA and also many private partners. So this is moving very fast. So in terms of the expanded access program, so this is something that NIH is taking the lead, but I really wanted to emphasize that expanded access is a pathway that is regulated by the FDA. And actually, the language here is from the FDA website, so I'm sure, Jacqueline, you're very familiar with it. But so expanded access is also oftentimes referred to as compassionate use. And it is a pathway for a patient with an immediately life-threatening disease, and of course, ALS qualifies here, to obtain treatment to an investigational therapy outside a clinical trial in cases when no other satisfactory or comparable alternative treatment option is available. And all of this, I think, is very applicable to ALS. So NIH has released 2 calls for applications, one in the spring of 2022. And based on that call for applications, we are now funding 1 expanded access study on a compound called trehalose in ALS. And then we had a second call for applications this spring. We've reviewed the application this summer, and we are going to make grant announcements over the next 2 or 3 weeks. So we are approaching the fiscal year, and we're all busy getting those grants ready to give to the investigators to start their studies. And our program really has 2 objectives. One is to conduct scientific research utilizing data from these expanded access studies. And of course, the other aspect is to provide access to investigational therapies for people living with ALS who are no longer eligible to participate in clinical trials. And I think that's a really important issue in ALS, because a large segment of the patient population cannot participate in clinical trials due to the inclusion criteria that are typical for ALS clinical trials. And usually it means you have to be relatively early in the disease to still be eligible to participate in a clinical trial. So a lot of people, they get diagnosed quite slowly, and by the time they really understand what's going on, they are sort of almost no longer eligible to participate in trials. So this is my last slide, just a few words about the public-private partnership that is being developed. And I wanted to emphasize, and Jacqueline already alluded to that, there are 2 components of this public-private partnership. So the critical path for rare neurodegenerative diseases was initiated last September, and that's a very close collaboration between the CP-RND Institute, the FDA, and other partners. And in parallel, NIH has been working with the Foundation for NIH to develop an accelerated medicines partnership in ALS. We are currently in the design phase, and here sort of we expect to both analyze and generate large-scale omics data and other biological data in ALS to facilitate and advance biomarker research, but also to identify new therapeutic targets and get a better sense about the causation of sporadic ALS. And what is really central to this effort is the standing up of an ALS knowledge platform. And we expect that this knowledge platform will both include high-quality extant ALS data sets. And actually here we have a working group with the Critical Path Institute and other members from the private sector to identify such data sets. Then, of course, they probably will need to be harmonized, but they will be uploaded into this knowledge platform so that researchers both in academia and industry can access them. But secondly, we are also going to generate new data. And this is going to happen through an ALS research consortium that NIH is intending to fund. We had a call for applications, we reviewed applications, and we are going to, again, make announcements for awardees before the end of the fiscal year. And this research consortium is going to provide a large-scale infrastructure for a prospective longitudinal natural history study in ALS, but also conduct biomarker research, perhaps optimize clinical endpoints, also do expanded access research, and sort of feed into this knowledge platform so that, again, scientists in academia and industry can access the data to advance their own goal. And another really important aspect about that clinical consortium is that they're going to collect longitudinal biospecimen from ALS patients that, again, will be banked in a publicly available repository to provide access to the broader ALS community. And I think this is my last slide, so I think I'll thank you for your attention, and I'll hand off to the next speaker.
Hi, good morning, everyone, and thanks to the organizer for the opportunity. I want to present today the advance in the AMP for ALS, together with a brief introduction on the FNH. So the FNH, or Foundation for the NIH, is not an arm of the NIH, but rather an independent organization funded by Congress to support the mission of the NIH. So basically, when there is the need to support the dollar fund with a PrEP fund from pharmaceutical companies and other organizations, then that's when we step in. Right now, we have over 125 active projects. Over the course of the year, we run over 600 projects and raise over $1.5 billion to support the mission of the NIH. The most famous, I would say, project run by the FNIH is ACTIVE, the recent COVID response to develop a therapeutic intervention, a vaccine, done a few years ago. But in the neuroscience space, I would say that ADNI, the Alzheimer's Disease Neuroimaging Initiative, is the most recognized project run by us, together with the AMPs. And I'll tell you a little bit more about that in the next few slides. So what is an AMP? The Accelerated Medicine Partnership started in 2014 when Francis Collins, at the time director of the NIH, was at a HIVR meeting talking with the executive directors and CSO of pharmaceutical companies. And we're discussing why it takes so long to develop a drug. What is the problem? What came up is that industry needs good, strong drug target data, together with good biomarkers to measure target engagement. And that was the inception of the first AMPs, that in the neuroscience space focus on Alzheimer's disease. Now we have AMPs in the neuroscience space focus on Alzheimer's disease, Parkinson's disease, schizophrenia, and now the ALS initiative. Overall, we have 10 AMPs, even though we are planning to develop a few more in the next few years. We raise, through this initiative, we ran over $800 million. We started literally 10 years ago, almost. In February is going to be the anniversary, with 34 industry partners, 16 NIH institutes, and 37 nonprofits working with us. What is AMP-ALS? This is the mission statement that was developed together with the People with Lived Experience and ALS advocacy companies. We'll focus on advanced drug discovery, development, validation of biomarkers and clinical outcome. I'll tell you a little bit more in the next few slides, and new therapeutic drug targets. We are right now in what we call design phase, and we're working very closely together with the NNDS, the FDA, and the Critical Path Institute, together with all our pharma partners and advocacy organizations. And the co-chairs of this initiative are Emily Gubitz, that is here, together with Steph Redet, that I'm not sure is in the audience right now, but will come probably later today. And then Dr. Off, that is the founder of Target ALS and individual living with ALS. What do we want to do through this initiative? As Emily mentioned before, there are two components of this initiative. There is the harmonization of existing data that is mostly on critical path, and then there is the generation of new data that is mostly on the FNH. And we are working very closely to be sure that we don't reinvent the wheel and we don't duplicate efforts in all this initiative. The first thing that we want to do is harmonize all the existing data. As probably you know, there are many data sets and initiatives focused on ALS. They're all valuable, but if we work all together, it's going to be just much more impactful than each organization can achieve on its own. So the first thing has been identify all these initiatives and all these data sets, and then bringing them all together. And then on the other side, there is the generation of new data through several omics, and this is just some examples of what we can do. We have 5 working groups right now. The first one is the portal and data architecture working group. And the goal of this working group is to decide what is the provider of this initiative, what is the portal that we're going to use to not only collect the existing data, but also to include all the other potential omics data that we're going to generate. There is the Extend Data Working Group, and this has been led primarily by Critical Path together with the NIDS to identify existing data sets. And then there is the Biomarker New Clinical Data Work Group, which represents the core of the initiative we can do at the FNH. What are the biomarkers that we need to identify? What is the work that should be done? And since it was mentioned earlier today, I want to add that some of the work that we're doing through this working group, including digital outcome, digital tools that we can use as well. Then we have the clinical endpoint working group to identify gaps in better certification of patients and disease phenotyping and phenoconversion. And this is managed by Critical Path together with us and the NIDS. And least but not last, we have a working group focused just on people with lived experience. But again, we have individuals with lived experience in every single working group, including the co-chair of this initiative. And I'll stop with a timeline of this initiative. We started not that long ago, and right now we are in the what we define design phase to define together with the industry partner what are the priorities that we want to work together. We're going to try to wrap this up by mid-October, and then we're going to start with the implementation phase. And the first outcome of this initiative is going to be the establishment of the AMP-ALS portal. That should take about 6 months to be completed, and then a few more months or years to bring in all the data sets and resources that are out there. And I want to add that all this initiative is in the pre-competitive space, so any pharmaceutical companies can access and should access all the data that we will produce. And with this, I'll stop. Happy to take any questions and talk with you now and later. Thank you.
Okay, I'm going to give the anchor talk, and then we'll open up for questions. I've been fortunate to hear from these, and you'll hear a lot of the similar voices here, because I can tell you, each of us talk, I would say, at least 5 times a week. So it's been quite a bit. Just to remind everyone, Critical Path, our model is built on bringing folks together. I mean, we are a convener, a neutral stakeholder, trusted partner. Our goal is really to identify unmet needs and looking at drug development and bring the best science and consensus to understanding how best to move forward through degeneration, as well as drug development tools. We work closely with regulators to make the non-competitive drug development tools available to the public community so that they can use those to advance therapeutics development. We undergo our submission to all regulatory endorsed with the hope, again, that they can be readily on-ramped into drug development perspectives. Just to give you an idea and a reminder of stakeholders, obviously regulators, the patient and person with lived experience community, nonprofits, industry, and academic researchers are really the cornerstone of these opportunities and efforts that we lead. CP-RND, just to remind everyone, Jacqueline alluded that CPath was selected to establish a public-private partnership with working with the various stakeholders, persons with lived experience, regulators, NIH, FINH. But I want to draw attention, because we hear this a lot, is this only ALS? And it's not. We've been charged with working within the ALS space, but also in the broader community. And so as you hear a lot of discussion in this conversation around ALS, CPath is committed to moving into multiple areas, and I'll talk about those in a minute ago. And we feel that there's great learnings that can be done by engaging other rare neurodegenerative diseases, because some of the questions are similar, and some of the learnings can be shared across different rare diseases. And so when I joined, One of the first initiatives that were done is we moved our ataxia programs and Huntington's under the same umbrella as our CP-RND, largely because they were more established and had learnings that we could draw from. The stakeholder community was well established, and that there are many things that we've discussed with those teams as they were brought on board. Obviously, the ALS initiative, and then we have others that are being brought on through task groups, and you'll hear that throughout the remaining of the time here at this meeting. But think of task groups as sort of directed, specific, early proof of concept areas of focus that CPATH undertakes, almost like a bit of a pre-consortia. We have one that's launching right now in PSP. We're targeting one in rare epilepsies and frontotemporal dementias in the beginning part of next year. Obviously, one of the areas that's important to us, and I stress this a lot on our calls, is that we're accountable to the public. So persons with lived experience are critical to this, and they're embedded in all the things that we do. And again, one of the beautiful things about R&D as a model is that we have the shared learning of multiple rare diseases that we can bring to the table to address common questions that we see. I put this slide in as a reminder, because like I said, a lot of the conversations that Alessio and we started about the same time was, how do you all know that you're not duplicating what the other people are doing? And so we kind of came up with some ideas around this, is that, as you heard, there'll be a lot of preclinical and discovery and translational science focus for FINH and NIH, whereas FDA and CPATH will be focusing not just on the, some of the review of existing data, but then how do you apply that existing data and new gaps to things that will accelerate questions around regulatory science. One of the things that's integral to this model is the shared connectivity. I'll give you an example. Alessio mentioned the extant data working group. CPATH had done landscaping around where all the data sources were. We brought that to the team. We talked about high levels of prioritization, where we could go with it. So the idea is that we can learn and share cross-functional gaps discussions. And so, our future state will be really looking at, okay, what knowledge gaps that exist, bringing those to stakeholders, be it persons with lived experience, academics, pharma, and then coming up with solutions, either it be through drug development tools or through new RFPs that discover new science. And so this model, we feel is really set, and it sets up a beautiful orchestra from as we've talked about this, because we can have regulatory discussions and the impact of that very early on in the design of NIH's science. So we're excited about that. Now, this is a busy slide. I'm going to go high level of this so we give some time of questions. What I wanted to point out here is a couple points of just what have you been doing in this year. CPATH, if you look at after first quarter, sort of the team that's been here really started in January for the most part. I can tell you we've moved really fast because like Alessio and I started at the end of last year, and we're at the state where we've launched working groups and are ready to roll into scientific plans right now. But what we ended up doing at CPATH is we did a lot of community outreach. We did a lot of trying to understand, where the unmet medical needs were for specific groups in both ALS, but other rare neurodegenerative diseases. We spoke with industry, understanding what their pain points, where did you cry after Type B meeting, and things that would really help us say, okay, what can we produce there? We worked in trying to, the ataxia and HD risk program moved to leadership, and they've been working out through a scientific plan, so there's been a lot of restructuring, and there'll be meetings during this time that really flesh that out in more detail, but a lot of activities of really re-level setting efforts within the rare neurodegenerative disease at CPATH. We've been actively participating in the AMP for ALS design phase and working on multiple work streams, and then co-leading efforts within the extant data as well as the endpoints and COA working groups. What you're going to see now is in sort of the next phases of this, we're both sort of working up to finish our scientific plans, which we'll be vetting to stakeholders, so that you're going to see towards late this fall and beginning of next year just an explosion of activity around ALS as well as the other neurodegenerative diseases. This just tells you some landscaping things, and I'm not going to go over great detail this, so we give plenty of time for questions, is that, one of the parts that was interesting about CPATH, as the R&D initiative stood up, is that we do have nonprofits and persons with lived experience engaged within other parts of CPATH, but R&D was going to take this to build on this, and we hired Tom Hart on really, and he's built a lot of interactivity and connectivity to the patient's community in ways that we hadn't explored at CPATH. And we're working on now to try to build capacity in this space so that we can really grow interaction and connectivity within patient-informed drug development within R&D and expand that further within the rare disease programs at CPATH. Data landscaping, I can tell you the number of data points we hit for this, no pun intended. We identified 62 plus data sets internationally. We interviewed most of the stakeholders working on data dictionaries, everything from what their architecture is to how many people, what are their biosamples, very great details. Those were summarized and prioritized and shared with the AMP programs so that we could begin to start talking about how do we best align for how they can be incorporated into a larger workspace and data warehouse. COAs, we participate and co-lead that working group. We were fortunate to have people like Michelle Campbell and Cheryl Kuhn, really also provide additional advice as we work through this space, because as you heard earlier today, the ALS-FRS is posed with some challenges. What we wanted to do is begin to take a larger view of ALS in that, would the concepts around and the measurements that were needed as we build capacity and things that impact people at different stages of their disease, as well as symptom domains, were addressed as we begin to expand the level of explosion in therapeutic development in that space. One of the things that CPATH does, if you know us really well, is we do a lot in quantitative modeling. We had our QuantMed people look at existing models and data that was done, so how they could be strengths and weaknesses and applied to regular science, and using AI as well as tools that could be further expanded in that space. And so we've already started looking at, from initial data sets we've had, applying new models and things to them already. Obviously, you've heard about digital health technology. This is one that's very near and dear to my heart, just because I think, one of the things that I heard significantly as we were doing our outreaches was the patient community really wanting access. Access was really stressed, and they felt that that was one of the venues by which they could be accessed. The second part of it is that using DHTs as ways to inform natural history studies so that therapeutic development could be inserted much earlier in the conditions. Now, our next steps, some of these I'm just going to say to be determined because they're in other sections at this meeting. So the ALS Consortium launch, we're planning finishing up this end of this year, and we're launching into the first January, February 24. We'll be talking about the focus group areas that we're working streams, and we want to hear back from prioritization in the session later today. HD Risk will be talking about their scientific plan, I think, and that does post-meeting discussion, so I won't steal their thunder there. CPTA has been really working on focusing on a staging system. One of the things that's beautiful about staging systems is they help with trial planning, but understanding, and setting the foundational steps for modeling. And they'll also be looking at trying to look at endpoints, specifically earlier in disease progression for the spinal ataxias. We'll be working a lot to expand the model of patient-informed drug development, and I want to thank those who are tuned in that couldn't make this meeting. I know that your time and investment and attendance is greatly valued, and we're glad you could listen in. And we feel very fortunate because many of you that couldn't be here today have been actively meeting and working with all of us on this stage and others in this audience. And so we're thankful for your time and your commitment to us moving this initiative forward. As I mentioned, we're not done yet. R&D will be actively looking at new areas, so reach out to us. We want to. It's good to have the problem of too much to do, and so one of the things that we really want to hear from the rare neurodegenerative communities is, where do we go next? Because one of the things we were hoping to do is hold a meeting in the first quarter of next year to identify new areas and develop project plans. And this will really work with the connectivity with RDCA-DAP because as you'll see, they'll be incubated in that system and connected to the CP-RND programs. And so we're committed to a research roundtable after that next steps. And I will stop now for questions from the audience. Go back. And before we have questions, I want to acknowledge some folks in the room. For those of you who are working in rare neurodegenerative diseases, can you raise your hand or stand up or something? There's a lot of people in this room. We're all committed to making this better. I can tell you I'm very, very excited about the colleagues at We Are Pro because we're all very passionate. Egos get checked at the door, and we're all just roll up our sleeves and get this thing moving. So we're excited, and next year you're going to be hearing more and more from us as we begin to make momentum in this space, and we're definitely be in touch with it. So questions from the audience.
Hi, Colin. Andrea Pauls-Backman, ALS Strategy Consulting. I've been involved in this space for 10 years. I've never seen this level of cooperation among the various government organizations, the regulators, and I just want to say I'm really thankful for CPATH getting involved, especially in the ALS space. So my question is this. Given that you've done this work in other rare diseases, what has surprised you the most? And you and I have worked together a little bit over the last few months, full disclosure. What has surprised you the most in coming into the ALS space?
For me, I can say for me, I'm fairly new to the ALS space. I can say with the things aren't necessarily dissimilar, and I think we've talked about this in some of in other meetings. But one of the things to me is that there's a lot of activity here, and I think that there's a lot of passion from the individuals here. I think the challenge for us now is really connecting the dots. So like making sure that you're doing this, you're doing this, you're doing this, and you're doing this, well, along with the goal of, you look, pediatric cancers, for those, pediatric cancers excelled because of their willingness to all come together along the same trajectory, and it resulted in really prolonged lifespans for pediatric cancers. And I think that ALS, what I'm seeing now is that there's more stuff, more data and more things here than I think I would have originally said. But I think now is just the putting it all together and us, kicking the blocks off the tires and getting moving. So, Emily, were you going to say something?
I mean, the question was for you, but I mean, when I think about ALS, I think one thing that is important to remember that to think globally about ALS, and there are so many excellent efforts in Europe, in Australia, in Japan, and just so always. And I think that was sort of one of the surprises when you guys started collating the high quality extant data sets. There are so many international studies, and sort of trying to sort of now tap into them. I think it's a huge opportunity, especially since some of the efforts, they're also relatively new there. So we need to connect with investigators, align in terms of what they are measuring, how can we do it in similar interoperable ways. So, I mean, that's always like something that I really want to remind myself. It's a rare disease, it occurs globally, and there's a whole sort of international community.
I want to echo that. That's another thing that's you're going to hear as this moves out is that I think the vision of this will be more global. And so, there's in our minds, we've already been talking to international groups. You're going to hear that in conversations. So others that want to?
Another comment. What surprised me the most is that the field is very fractured, as you know, but every single organization has been so nice and easy to work with. And I asked the question, why is the case? And it seems that this is the time. So as we are working all together, all these foundations are coming together as well. And so this initiative, I really hope that it's going to be successful because literally every foundation has been supportive and collaborative.
Well, thank you. I want to say, I said something to Emily earlier this morning. We're at a place in the ALS landscape where for the very first time, there are funds available, significant funds available for research and for programs like these, and we have the coordination. So those two things are key. Thank you.
Thank you very much. I'm Kathy Collett from Indianapolis, Indiana, a longtime ALS advocate. I love what I hear, and I'm most appreciative, but something is bothering me in the back of my head, so while you're all here, I'm going to tell you what's bothering me. We saw FDA grants for natural history studies, plural. And it's like, I get it, but at the same time, I'm thinking we need a honking big natural history study, and we need to get all these other natural history studies together. And then I hear about harmonizing data. And I think harmonizing is a word that is not well defined. And I think we have a lot of people doing a lot of good ALS research. And when they hear harmonizing, they're like, good, you'll make my data fit with the rest and you can use it. And I know the word standards is not something that creative people like, but as a part of this, can we come up with some standards so that people are actually generating data at the source that make the data much more combinable? Because today, there's no doubt we have terrible silos. So thank you if you can reassure me.
Do you want to take that with?
Yeah, certainly, that is something that we think about on a daily basis. There are common data elements for ALS that were developed by the community, by NIH, but it's still sort of a little bit difficult to implement them. But in terms of the natural history study that FDA is now funding and synergizing effort that is moving forward to NIH, we are going to talk, and we are going to talk with the investigators. There'll be overlapping sites, so it's not that we sort of do things in silos. Right. This is sort of more of a continuum. And natural history studies also look a little bit different in terms of what they want to focus on. And there are different outcome measures, and maybe one natural history study will sort of emphasize the collection of longitudinal samples for high new omics technologies, or another one they want to go more into the direction of digital outcome measures, things like that. So, but what is really important that we'll be able then to combine the data and sort of make it a stronger, bigger study, exactly. Just up the sample numbers, as long as we ensure that the data is collected in the same way. And it's a little bit more difficult sort of thinking about older studies that, were performed a few years back. But even then, we already had some data standards, but it's something that is sort of we're thinking about every day. And it's, I think the investigators are very much aware of it.
Yeah, and just to add to that, that's something I think that we're in an opportune time, because sitting in this room, I see a lot of people that do data standardization all of the time, and they make it for multiple uses. And I think that the beauty of this now is that we have the ability to connect this and speak to this from a broader community. And I think if we're going to get there, the time and talent and treasure is all in this ecosystem now to make that happen. And like I said, the other part is feedback from people like you that keep us honest. Okay, last question from the audience here.
Hi, I'm Frank Serwick. I'm Chief Medical Officer of Spiro Pharmaceuticals. We don't do ALS research, but I've been very involved in critical path and other areas for many years and have watched the evolution. And I'm really thrilled and congratulate you on how close you are to the pinnacle of collaboration and cooperation today as compared to where we were when it wasn't Critical Path Institute, but Critical Path Initiatives, one separate for NIH, one separate for FDA, who didn't talk to each other until the patient foundations actually brought that together in a public-private partnership. So the one comment I have or question I have is sort of addressed in terms of the about to collaborate and discuss the work that each agency is funded to do. I would just encourage NIH and FDA, which you've said you already are going to do, even as I was standing up here, to talk and to learn from each other, particularly from the perspective of the experience that we had in autosomal dominant polycystic kidney disease, where there wasn't that communication. And so NIH developed a very expensive and long program to develop an endpoint that they hoped would be useful for drug development. It actually was. It ended up being very helpful, but it took an evolution, and it took that bringing together. So I would encourage you that have an NIH reviewer or someone speak with you about your programs in terms of the clinical research that you're going to be funding, and vice versa. Have someone from FDA very involved in the continuing clinical research you're doing here and in other areas, so that there's an understanding of both the basic and practical science that's needed. Thank you.
Well, thank you. Thank you, everyone, for this. For the being able to attend the session. Sound? Can you hear? Okay, thank you. That will conclude this session, and then we will go ahead and pass to the next.
Good morning, everyone. My name is Sept. I'm a clinical team lead with Rare Disease team at OND CEDAR, and I will be talking about Rare Disease Endpoint Advancement Pilot Program, which we call it RDEA. It's a program, it's a joint program between CEDAR and CBER at the FDA. We talked about rare diseases, and as we all know, millions of Americans suffer from rare diseases, and there are not approved therapies for most of those individuals. And the reason are, as discussed all day, are the challenges that come with rare disease drug development, including the poorly understood natural history of the disease, the nature of these diseases being serious, life-threatening, progressive diseases. And a lot of these conditions involve pediatrics, and that is another unique challenge in rare disease drug development area, all the difficulties and challenges that come with drug development for pediatric children, pediatric population. Small population of the patients are another challenge with these diseases. Diversity in phenotype and genotype of these diseases, again, add to the challenges of rare disease drug development. And lack of development tools and lack of outcome measures, including biomarkers, and lack of solid translational science background. All of those add to the challenges in rare disease drug development. And one of the other challenges in rare disease drug development include the lack of clinically meaningful endpoints. That brings us to the Rare Disease Endpoint Advancement Program. RDEA pilot program is a joint CEDAR and CBER program. It's a PDUFA 7 commitment that we have implemented. And the goal of the program is to advance rare disease drug development by providing a mechanism for sponsors and stakeholders to collaborate with the FDA throughout the efficacy endpoint development process. The program opened for submission in July of 2023, which is the fourth quarter of 2023. We will be accepting one proposal for this quarter, and the submission ends September 30th, 2023. In the following years, we will continue to accept proposals into the program, 3 proposals per year, which is roughly 1 proposal per quarter. And FDA goal is transparency. Our goal is to share the information and the experience that we gain through this program with the public, and it could be in different ways. It could be development of guidance documents, public-facing websites, or public workshops, which we had our first RDEA pilot program public workshop in June of 2023, and that is publicly available on our website, and we will have hopefully 2 more workshops by 2027. I'm briefly going to talk about the eligibility criteria for these proposals. The first eligibility criteria is having an active development program under an IND or pre-IND. There are some exceptions there. We understand that some natural history studies may be exempt from IND or pre-IND, and those will be considered for admission to RDEA pilot program if they meet other requirement criteria. They should be addressing a rare disease. Again, another exception there is if the endpoint proposal is for a common disease that could be used in a rare disease, could be applicable to a rare disease if the sponsors provide adequate justification on how it's applicable to the rare disease, they may be considered for the program. And the other eligibility criteria is that novel efficacy endpoint should be intended to establish substantial evidence of effectiveness for a rare disease. For the RDEA pilot program, we define novel as an endpoint that has never been used before to support a drug approval, or it has been substantially changed from its previous use. Once we receive these proposals, FDA will send an acknowledgement letter to confirm receipt of the application, and then all of those submissions will go through a review process, eligibility, completeness process, and a selection process, which is a very collaborative approach between CDER and CBER, multidisciplinary review. Depending on the type of endpoint that's being submitted, we will consult other subject matter experts. For example, if the endpoint is about a surrogate biomarker, we will consult our colleagues from Office of Clinical Pharmacology, or if it's the COA, we will consult those SMEs in digital health technology or other types of endpoints. It will be a very collaborative review of those proposals to choose one final submission per quarter. 60 days after the end of each quarter, FDA will notify the sponsors about the status of their proposal, if they've been accepted to move forward or if they've been denied. And then we will go through a disclosure discussion. As we discussed, the goal of the FDA is that to share the information that we learn through these programs. So it's very important that sponsors pay attention to the disclosure elements and be aware that their proposal may be discussed through different public venues. And how do I go back? Would you please go one slide back? Whoops. Okay, the required information for these proposals include executive summary should be 1 to 2 pages, and a total of proposals shouldn't be more than 12 pages. They should include all of the required information, and if the sponsors believe that there is any additional information that will be helpful to their submission, they may add those to their proposal and to their submission. I'm going to go quickly through these required elements, including the product name, IND or pre-IND number, proposed indication. If the proposal is for a natural history program, any type of information required for that natural history, including disease epidemiology, additional information that the proposed natural history will add, how the design of natural history study provide this endpoint selection for future studies, and also proposed timeline to design and conduct the natural history study should be provided. If the proposal is for a common disease, there should be enough information about how the endpoint will be applicable to a rare disease. There should be adequate justification that the proposed endpoint is a novel efficacy endpoint intended to establish substantial evidence of effectiveness for a rare disease. And also another required information is about how the endpoint will measure a meaningful clinical benefit for the patients, and also detailed description of the endpoint, what type of endpoint is being used, and how it will be verified, how it will be validated. Another piece of information, again, as this was discussed, it is very important to consider patient and caregivers' input. We have a very limited resource of information with rare disease space, small population size. It is very important to consider how the patient input will be included in the studies, and for the sponsors to include that information as well. A brief history of the development program is important to be included, overview of the study design, objectives, statistical plans, and elements of the endpoint novel development program that are non-disclosable. Again, those should be included in the proposal. And a list of the questions that you may have with the agency. Proposals that are accepted, that are admitted into this pilot program, have the opportunity to have up to 4 RDEA meetings with the agency. Similar to other meetings, once the sponsors submit their meeting request and a complete meeting package, the meeting will be scheduled within 45 days. And these meetings, the RDEA meetings, are totally separate from other type of meetings that the sponsors may request with the agency, including IND or pre-IND meetings or other NDA or BLA meetings. Those could be done around the same time separately, but these RDEA meetings are separate and are in addition to other type of meetings that could be provided during the drug development program. And these meetings are focused on efficacy endpoint development. So if there are other questions, sponsors may request other type of meetings for their programs. The information that is being exchanged and the discussions during the RDEA pilot program do not constitute a regulatory decision. Those are non-binding, and they do not guarantee approval of a product. If I can go to the next slide, I think. We have multiple resources at our FDA's website, all type of information for the proposals, for the meetings, deadlines, everything is there. Our email address is included there if you have any questions. If the program is for a natural history study, again, we mentioned that they they do not have to have an IND number or pre-IND number, but there are some requirements for our tracking purposes. They may request a pre-assigned application number, and the information all is included on our website. And I thank you for your attention. And I think that is my last slide. And if you have any questions, I'd be happy to answer. And I believe it's...
Thank you so much. Okay, thank you again. So now we are going to go ahead and take a break for lunch. So just on the outside of these doors, we have lunch set up in box lunches. We have about a half an hour for you to grab your lunch and then make your way to the showcase room. So you can grab your lunch and come back in here and sit down for a few minutes, or you can grab your lunch and go ahead and take it to where the showcase sessions are taking place. And we have staff out in the hallway that will be directing you. We have 2 showcases on this floor, so one here at the end of the room, another one down at the other end of the hall, and then we have 2 rooms on the 3rd or 2nd floor, which is 2 floors up. But we'll have staff directing you, so we can't wait to see you guys at the showcases. Enjoy your lunch. Thank you.